So as the pilots are getting set up here, we'll just give you a reminder of our current situation. This is the Voyage to the Ridge exploration team coming to you live, streaming over the internet from the NOAA ship Okeanos Explorer. We are about 340 kilometers north of the Azores Islands on the Mid-Atlantic Ridge, and we're diving on a feature today that we have named Zenith. As I've just noted, we're coming in at about 1,075 meters, so just a little deeper than 1 kilometer. And we're on this topographic high. It's a ridge, or you can call it a seamount, if you will. And we'll be climbing upslope throughout the day, looking at the communities that are associated with the hard bottom. And I can already see 3-4 fish in view here, so that'll be interesting. And I noted as we got to within the last 40 meters or so of the bottom, there was certainly a pickup in the amount of organisms that were in the water column. So very interesting, hopefully productive dive today. And we'll have a few minutes here as the pilots get situated, and then we'll be ready to start requesting zooms on some of the organisms we're seeing, and we'll start heading upslope. So stand by for that. Copy that. Thank you. Our first step, we would like to take our second Niskin sample before we start to look at some of the fish around here. Thank you for that, pilot. So there's several fish here of the same species. You can zoom in on the one that is closest or most convenient for you.
Hi, Scott. This is Tom from the Control Center. We have seen some aggregations of orange Raffi. We think that this might be an Oplos etus atlanticus. A general information for you guys is that the Azores back in 1995, I guess, introduced a trawl of orange Raffi. So when international companies wanted to start coming to the Azores to do explorations of orange Raffi and Okeanos, the Azores decided to ban bottom trawl. And that's probably one of the reasons that we still see a lot of orange Raffi in this area. Just a comment that could be of interest for the general audience. Thank you.
Absolutely, yeah. Thank you for that, Telmo. I'm curious, do you know if, I guess we wouldn't know if on this specific feature there was any previous fishing, but we might see evidence of it once we get on the top, so that'd be interesting. So Telmo's saying these are orange Raffi for which there was a fishery, and is one of the reasons there's a lot of interest in protecting some of these areas. These are very slow-growing, long-lived fish. And we can see there are several here in the area. And I'll note, Telmo, I don't know if you saw it, but I did see at least one Neocytus, the Oreo, when we were setting up on the bottom there. So that's great, Pilot. So let's have a look. Let's go wide. I noticed on the left as we were coming over here, there was kind of a wiry coral sticking off of one of those rocks in the bottom. And Ashton, I don't know if it's too soon to say anything, but I'm seeing a lot of boulder rubble here if you want to make any comments on.
Yeah, thanks, Scott. Still waking up. Yeah, so we see a lot of basaltic rubble, kind of similar to what we saw in our previous dives. You know, some of these larger features, like the one you're looking at just now in the center frame, look like it could be in place, but really hard to tell just given the large amounts of rubble. You know, I imagine a lot of these probably have a manganese coating on them. So manganese is a mineral that precipitates out in the deep ocean. To give you some context where we are, we're about we're probably about 15 kilometers east of the Mid-Atlantic Ridge section that's on our left, and we're kind of just above a small transform fault system that is a step over in the Mid-Atlantic Ridge. It's a small fracture zone that's about 15 kilometers wide, where there's shearing and there's some faulting that causes the Mid-Atlantic Ridge to step over from its north-south direction 15 kilometers west and then continue north-south again. To give you some context, the seafloor that we're looking at, the age of the seafloor and the oceanic crust we're on right now is probably about 1.5 million years to 1.3 million years old. So fairly old, and it's given a lot of time for things like manganese to precipitate out.
Pilot, let's have a look at the coral. Well, go ahead, the one that's straight ahead, but then the farther left, just on the edge of this rock field. Sorry, Pilot, I'm not sure if you heard me there. I'm not sure if you're on the OKEX line. Yep, copy that. Yeah, thank you. I just wanted to be sure to push your widget here. I'll also notice that there is all these sort of whitish blue circles on all of these rocks. Those are all sponges, encrusting sponges, so there's clearly a lot of sponge life here. And for the scientists on shore, one of the rocks that Ashton picked up yesterday from our axial volcano dive, I was able to peel off one of these small crusting sponges. It was only maybe 1.5 centimeters, 2 centimeters in diameter, but it came off in pretty good condition. So this looks like a stony coral, Sclerexsonians, probably Lophelia, as we're focusing. I'm pretty certain that that's what this is. And in some parts of the North Atlantic, these Lophelia form pretty substantial reefs that have grown in place for tens of thousands of years and are home to just an incredible diversity of other organisms on it. Here I can see very wispy in the foreground, there are some hydrozoans growing. This coral is alive. I can see the tissues coming out of some of the calyces. That's a little cup. For some of them you can see face on. They have these white lines in them. Those are called septa. And the polyp, the tentacle part, can kind of retract down in there. So you see that soft pink color. That's the live tissue. And where the skeleton is much thicker, That's probably the result of a worm that is living in association with this coral. And the coral has actually formed this tunnel or garage around it. Thank you. Good close-up. See a shrimp crawling over the rock there, and then on the surface of the rock I'm seeing some tubes with sand. That's probably also polychaete worms, annelid worms. Some yellow sponges in the background. I'm already, I think, seeing some black coral that Telmo suggested earlier we might see. So yeah, hard to the left, please. We'll get to the black coral a little bit later. That's the orange-looking corals that we see in the foreground. But I did see a planar fan over here to the left. Should be coming into view in a moment. It's further back and left still. And I see in the foreground there a little purple plexaurid unbranched, which is kind of interesting. No, I think it's behind us a little bit. Yeah, you're probably right over it now. Pilot, if you could put the lasers on also while we're moving. Thank you. So the lasers are on there now. There's a Leopathes black coral in the foreground. I still don't quite see this planar fan that I saw, but we should be right over it. There it is. Just came into view, lower center. I'll also note that I think I see a cup coral on the rock to its left, and I'm kind of noting that for the folks on shore. And on the far right, you can see a dark, bright black line on a rock. That suggests to me that there's probably something like a chitin that is feeding on the rock and leaving a feeding trail behind it. So you can see the two red dots there. Those are lasers that are mounted just above the camera. They're 10 centimeters apart, so they allow us to scale. This looks like it's another, a nice pillow feature that this coral is sitting on.
Yeah, it looks like a large fragmented pillow, and you can see kind of it probably, when it was being in place, the lava was actually flowing from kind of the bottom left to the upper right, and so that place where it's broken off on the left-hand side is probably the direction it flown. It's too early in the morning, and then kind of stopped and then formed that bulbous section on the top right.
Got it. So this looks to me like a coral in the family Anthothelidae. It's a Anthothelidae, excuse me. It is an octocoral. You can see there very nicely 8 tentacles, hence the octo. Each of those tentacles, there's a little side branch. And what I'm looking at is the texture of the tissue, the density of the tissue, and just sort of prior knowledge. I can see some grooves along the axis. So I believe this is the coral that we collected on dive 1. We weren't certain what it was, and when it came up on the ship, it was pretty clear it was an anthotheelid. You see there's an orange ribbon on this branch on the right side. That's probably a Nemertean worm that's crawling on the coral. So that's kind of cool. Many of these corals, of course, have associates. I'm seeing in the lower right, looks like a brittle star on the backside coiled around there. Thank you. That's a good view of that coral. There's just so much to look at around here. But we can sort of move towards upslope, and I'll identify things in front of us. It looks like there's no shortage of opportunities to look at things. Yeah, so I'm already seeing a couple of the black corals that Telma noted in our introduction. They had found on some other ridges up here in some high abundance. And these Leopathes black coral are particularly slow growing. There's a handful of corals geochemists have worked on to get some aging And they usually do that using some stable isotopes of the skeleton. Some of these corals are able to lay down the skeletons in layers, and so you can basically interpret how fast those layers are being deposited and extrapolate to get an age. I have to open my notebook here. I was making some notes as we were cruising out here on aging. So some but not all corals show growth rings in their skeleton, and for at least some of these species, the growth rings are annual. And some examples of that are Okeanos arbuscula, which is a bamboo coral, Primnoa ricidiformis, and then the black corals, Storopathes and Leopathes. Let's see, I have an age estimate for Leopathes from the Gulf of Mexico. They've been aged at 300 meters depth from 530 to 2,100 years old. And the oldest one known come from off of Hawaii. There's a couple, one was aged at 2,377 years old and another at 4,265 years old. Really quite amazing. And we'll talk a little bit later about what that means for their rate of growth. This is a sponge that we're looking at in the foreground. It's a glass sponge of some kind, and I think it's mostly dead from what I'm seeing, that brown tissue. And, actually, if we could look just to its left, that little colony that's on the rock, I believe that's an Okeanos. And there's a reason I want to look at this one closely. I'm pretty sure I know what it is, but it's a bamboo coral, and this is a very interesting species in that it is able to grow on both hard substrate, like this rock you're looking at here, and in soft sediment. And what it does is it alters the holdfast at the base. So I can already see there's a pretty wide white holdfast, which is the base of the skeleton that's glued to the rock. And when it grows in sediment, what you want to picture is wax dripping from a candle, not the white one, the orange one to its left. My apologies. That was a nice zoom, though. When it grows in the sediments, that holdfast drips kind of like wax, and this is the calcium carbonate that's being secreted, and it creates these tendrils or these roots that go down in the sediment and allow it to be stable there. Excellent. Yeah, so that's Okeanos arbuscula. So this is probably one of our better views of the bamboo coral, which we've noted in several of the dives. And you can see clearly here that it has a skeleton that alternates between those white, what are called internodes, which are primarily calcium carbonate, and then the brown lines, those are primarily protein, and we call those the nodes. And it's that alternating appearance that gives it the resemblance to the bamboo, hence the common name bamboo coral. And this genus Okeanos, you notice that it's growing as a small bush, and that is that the branches come out in multiple planes. Thank you for that. That's a good view. And looks like there is a squat lobster in there. You can see one eyeball and two long arms, and I think, I'm not sure if the pinkish mass towards the center are eggs that it's carrying, or if there's an anemone in front of it. But very often, if we go a little bit higher, is that an, no, it's a second one. So there's a second squat lobster facing us. You can see another eye. It's kind of black with an orange spot in the center, and it's two long arms. So these are chirostylid crab squat lobsters that are specialized for living in corals. So a lot of these corals have very specialized hosts. Associates, excuse me. The coral is the host to the associates. Very nice. And when video's done, we can move on. Manuela Ramos on shore suggested the first sponge, that large sponge just to the right, we're going to see it again here in a second there, may be a Hertwiggia falcifera. And Manuela, I'm sorry, I'm not sure which isidid you're referring to for the collection. No, I'm chatting with onshore. Thank you. Yep. There's a couple of fish in the background. They're pretty far for a zoom, so let's have a look at this large fan-like thing that's on the rock on the left of center. I think it's going to be a very large sponge. Oh, actually, it's a stony coral. It might be a couple of, oh, I see, yeah. So this is a stony coral with most of it's dead. And what I'm trying to see is, are there more than one? Or is it just one massive one where only the outer tips are alive? I think that's what's going on here. This may be in the genus Enelopsamia, another stony coral like the Lophelia that we just saw earlier, but this one is clearly growing much more like a fan rather than the sort of three-dimensional irregular shape that the Lophelia takes on. Yeah, Manuela Ramos is saying this is Enelopsamia rostrata. So stony coral, this is related to the corals that are forming the large tropical shallow reefs in the Caribbean and the Great Barrier Reef and so on. But there are about half of the known species of stony coral do live in the deep sea. They just don't tend to form the large mounding corals that you see. Instead, they're typically branching. And I can see there's a bunch of stuff tucked into the other side of this coral. So it's been around here for a long time. I really don't know that anyone's tried to age these Enelopsamia, so I can't tell you how old this is. I suspect it's pretty old. It's been around for a long time, but long enough that the lower parts of the skeleton are dead. It's a colonial organism, and so, you know, parts of it can die, but other parts are still alive. So that yellow that you see up there is still alive, and the polyps are all facing the other direction. Yeah, Pilot, I don't know what your situation is in tether and all that, but if we can get a view of the facing side, that would be helpful, but not critical. Correct.
We were going to first try to get you a view between the changes.
No problem. That's okay. So you can see we're looking closely at the surface here, and you can see a number of other organisms living on here. This is kind of one of the beauties of these close-ups. These corals are forming habitat for a lot of other organisms. So I'm seeing several squat lobsters. I see sponge overgrowing here. There's a small crinoid just left of center. I see a cup coral that's growing. I see several barnacles that are growing up there. There we go. Beautiful view of two cup corals, one much smaller than the other. The size is also telling me something interesting, and that is that we've had relatively recent recruitment. That is, the larvae of these organisms have settled on this coral and begun growing. So we have lots of different timescales being represented just by this one colony, the very long ago period at the base of this colony, and then more recent. There's actually... It's really small, but it's just above the cup coral, but there's a baby crinoid there. So crinoids like the feather star just above and to the right, on the screen now, the feather star, start stalked. And so just below one of those arms, you can see this little flower-like structure on the stalk. That's a baby crinoid that hasn't left its stalk yet. Nice. Another example of recruitment. So I'm not sure why parts of the skeleton are dead, if it's being grazed by other organisms. Actually, there's a snail also in the lower left. Or if it's just senescence, old age, that some of the tissue is lost. And let's see, you can actually see some of the polyps tucked into the calyces. They're sort of a darker yellow. And I'm not sure what that mass is that I'm looking at. Could be a nudibranch, possibly. Yep. That one's a mystery. And then that fuzz at the top, that brown fuzz, those are hydroids. Cool. So we have these sort of isolated boulders on top of this ridge that are serving as a substrate for a coral, which is itself serving as a substrate for a whole host of other organisms, and is one of the reasons why ecologists are so interested in trying to. Oh, and there's a interesting urchin on the back there. I didn't notice that when we zoomed in. Why ecologists are interested in preserving the corals, because if you can preserve those, you preserve lots of other elements of the community. Not quite as many pteropod shells as yesterday, Ashton.
No. Yeah, it actually seems less sedimented than the place we were looking at yesterday. Yeah. And it looks like we're looking at it looks kind of like a talus pile, but it might actually be more primary in situ pillow basalts. It's kind of hard to tell. There's lots of talus mixed in, but there are some larger boulders. And in the bottom left, you see a nice cross section right through that circular fractured area. That's a nice cross section right through the end of a pillow tube.
Absolutely. I do want to look at that pillow, but if we could look at the fish, just because they're mobile while we see them, if we can get a close-up on them. Not the red one, the one going out the other side, the eel-like one. My bad. We've seen that red one really well. We know what that is, so we don't need a lot of zooms of that. Thank you. Yeah. So there's two different eels here. There's the one on the bottom, and then there's this one we're going to look at closely. You choose one, whichever one's easier. Because we're going to say we want it all back here.
Now, I don't recall seeing this before, but...
Yeah, we may finally have a... actually, is this a duck-billed eel, perhaps? Every time I say it's a duck-billed eel, Mike Vecchione comes on and tells me it's a snub-nosed spiny eel. But he's not here this morning, so he can't tell me that.
Well, I can say with certainty it looks very Never-Ending Story-ish.
Let's see here. I'm looking at the fish guide that was sent to us. Okay, so it's not a duck-bill eel. I can rule that one out. And it's not a conger eel. I don't think it's a cutthroat eel, Synaphobranchid. Oh, it could be a. It could
I don't
be a
know about that.
Synaphobranchid.
Is that red coral down at the bottom, or is that a Syngid?
I missed that video. All right, we'll stand by for a word from Ken Sulak or one of our other fish biologists on an ID for that eel. Thanks, pilot. That's good.
We got a good headshot?
I think so. So can we look to the left of the lasers is a bright white fan sticking off the rock just above the yellow one. Just a bright white one that I'm interested in. I'm wondering if that's a bryozoan.
The small one?
Yep, the small one. I'm trying to, get some of the different sort of stuff that we don't see all the time. This is a hydrocoral, a Stylasterin Hydrocoral. So it's a hydrozoan. It has a very stiff calcium carbonate skeleton, but it belongs to the class Hydrozoa. And then below it, that interesting ribbon thing on the rock to its right, that is another kind of octocoral, forming a very interesting shape. And then it's an octocoral like that. We've looked at a couple of the bamboo coral, all the sea fans, but these ones don't produce an upright axial skeleton. Instead, they grow like a ribbon or a mat. Several encrusting sponges on this rock. Let's have a look at the golden yellow one down below. Right in the center, the lasers are on now. This is yet another octocoral. This may be an Acanthogorgia. Yeah, I'm willing to bet this is in the genus Acanthogorgia, or at a minimum in the family Acanthogorgiidae. And the reason I'm saying that is you see they have tall polyps, and the tissue around the skeleton is really thin, so they really have nowhere to go. And if you had just the right angle, you would probably see they have characteristic what are called a crown of thorns, and that's the microsclerids that they have form this crown of thorns around the tentacles. Thank you, that's great. We're ratcheting up our diversity here. Tom will suggest that may be a Synaphobranchus, so indeed that would be a member of the cutthroat eels. That eel that we're looking at, still there on the right side. I think I just see another bamboo coral there as we move away. I mean, there's so much here, we can get overwhelmed and not go anywhere. So we're trying to get periodic close-ups to get some sense of the diversity. Just a reminder to everyone on shore, we can collect, but we're limited to 4 biological samples in the bio box, and then 5 slurp samples, and then 2 rock samples. And so we want to choose those samples carefully. I've already had word that given that we've seen up here different locations of these black corals being kind of abundant, and maybe, Pilot, if you're in a good position to go to the upper left, whatever direction you're going, you don't have to go back because there's lots of them. You tell me what direction you're going. Okay. There's lots of these black corals. I want to get a good look at one of them. So I don't want to try to change a move that you put in. I think I'm seeing up on the top of this rock several of the Acanella colonies that we imaged earlier. 1, 2, 3, 4, 5 at least. Anyway, I was saying that given the interest in the black corals and the uncertainty in what it is, that will likely be one of our collections today. Yep, just to the left of the lasers. I can already see that it has some squat lobsters in this one. So this is a Leopathes black coral that we're going to be zooming in on, and so that's the one that I was referring to earlier. Has been, as far as I know, the oldest known documented coral. In this case, it's a black coral. The black corals belong to the subclass Hexacoralia, and I say that only to separate them from the octocorals. So black corals are more closely related to the stony coral, like Enelopsamia that we saw earlier, and the cup corals and the sea anemones, than they are to the octocorals, which otherwise have similar sort of growth form in that they have a protein-based skeleton. They form these sort of plant-like appearances, tree-like appearances. Black corals as a group have tissue in lots of different colors. This one is clearly orange, but with Leopathes it's usually easy to see on the main axis of the branch through that tissue, and you can see that the skeleton itself is black, and that's due to this antipathin protein that is used to form that skeleton. In Leopathes, the polyps themselves are particularly small. I know from experience they produce incredible quantities of mucus, such that we could clip a small branch, put it in a container on the ship, and then you could essentially put your hand in it and pick it up like jello. And it's really interesting to think it's these small polyps that we're zooming in on. Each of them are producing this polysaccharide mucus that they emit, presumably for defense when they're bothered, and that absorbs water and expands. So if you want to compare this to the polyps, if we could just, right in this very center, yeah, that focus is perfect. So you can see that polyp right in the center against the black branch behind it. It has only 6 tentacles compared to the 8 that we saw earlier, and there's no side branches. So just one trivia fact for you to separate the hexacorals from the octocorals. Thank you. Pilot, so at some point today we would like to collect a black coral. If this is a convenient time, we can do it, but we could also wait till we're in more stable position. I'm seeing lots of them. So there are a couple of different species, Leopathes grimaldi and Leopathes expansa, and it isn't known which of those two species is the species that's dominating these ridges up here on the northern part of the Mid-Atlantic Ridge north of the Azores Plateau, and that's why we would like to have this collection. Once you have knowledge of the species that's here, it's easier conservationally to justify protection. Manuela Ramos adds that the squat lobsters that you're seeing on this colony, I see at least two of them, are probably Gastrotychus formosus. And those squat lobsters have an association specifically with these Leopathes species. Yeah, I think I saw this crab earlier. He's climbing up the slope. Let's see. It's a brachyurin of some type. So, Pilot, just to confirm, you are going to try to sample this one? Absolutely. Yeah, so we don't need too much, an outer branch. Let's try to leave most of the colony intact. Copy that. Yep. And just as a note, black coral skeletons are very hard, and so I think this will need to be cut. You likely won't be able to bend and snap it.
Science, I just changed stream 2. You may want to look at the HD2 camera. It looks like we have someone wanting to be sampled.
On shore, if you switch over to camera 2, you see that brachyurin crab that I was referring to. It's checking out our rock box. It's a pretty good image of it. I'm sure somebody can identify it if they're familiar with the crab fauna around here. Not sure that it's a Chasion, but it may be.
Okay. Video's full wide, full clear. I'm going to hold that camera in HD2 zoom for a bit. All cameras are full wide.
All right, on shore, so stand by for collection. We have to do some documentation notes here on shore, on the ship. As the pilots are getting set up, I'm looking at that colony, the white colony that's on the left side, just going out of the screen right now. I'm not sure what that one is, so we may have a look at that next. We're at 1,064 meters depth, and we settled at 1,072 meters depth, so we've only come up what is that, about 8 meters. We haven't traveled very far, but there's lots to look at around here.
So stream 3 has been modified for 4 different camera angles during sampling.
On shore, if you want to follow this collection more closely, you can turn over to camera 3, where the quad screen has 4 different cameras to show various angles, I believe including the it will later include the sample box. And you can see that this is a difficult operation. It's not straightforward. It's straightforward to ask for a piece. And then the final challenge is always stowing it into the bio box and not having it drift or float away. But the conditions look pretty stable here. My colleague Les Watling has added a note in the chat room that's kind of interesting to what I was saying about the age of Stylasterin before, and that is the one in Hawaii that was determined to be so old, a 4,000, what was in notes here, I think it's 4,200 and change. I guess once you get over 4,000 years old, maybe those last couple of numbers don't matter so much. 4,265, and he notes that it was determined to be the oldest living animal when it was collected, and not surprisingly, no one had the courage to get another colony to see if any others were equally old. Once it was understood how old these were, of course, there was all interest in preserving them. This colony is a little bit smaller than I was expecting, actually. It's always interesting, the perspective when you're looking in the in this 2D camera at this 3D environment. Yeah, that's a really healthy piece. And there you can see the caridoid response of that squat lobster, and they're flapping their abdomen and moving backwards, and that's a typical escape response for shrimp and lobsters that have that morphology. I foresee a lot of mucus in my future tonight. Yeah, Les Watling adds, the problem with the aging is that in order to get the accurate age, you need to have the base of the colony, which means you need to take a really big sample. And so we try to minimize the number of times we do that. Mike Vecchione has a really interesting question. He says, does old colony mean old animal? And I think what he means by that is a colonial animal is constantly producing new feeding structures. New tissue is being turned over, and I've thought about this a lot. So all of the tissue that you see on the colony now is probably completely replaced from when it first started growing. It's intriguing. And if I'm misrepresenting what you meant, Mike, please let me know. Yeah, this is a big piece. Looks like there may have been a little associate on that, not the squat lobster, but something else. There we go. We have that successfully stowed in the starboard inner bio box. Very nice collection. Thank you, pilot. That was terrific. Pilot, nice collection. When you are prepared, we're interested just off screen left. There's a white colony that's hanging off the rock and pointed downwards. Yep, just over the toe now. And then after that, just going off upper right, there's a barrel sponge. Thank you. James Aubrey, graduate student at the University of Louisiana Lafayette, whose dissertation is on Acanthogorgiidae, notes that the Acanthogorgia we saw earlier is interesting in that that would be the deepest observation in this part of the Atlantic of an Acanthogorgia as far as he understands. He says most of the species that are described relatively close by, at least in the eastern Atlantic on the coast, are from 240 to 500 meters. So possible species that have been described, some good names here. Acanthogorgia hirsuta, which means hairy, I think, horrida, and truncata. Thank you, James. So we're going to have a look at this white coral here. Might be different, and then we're going to go and look at the large sponge in the background in the chat room. Joanna Javier has joined us, and she's a sponge specialist. She spotted that thing right away when we were looking at the Stylasterin, and she will be your biology lead on EX2206, the follow-up expedition after this one. And I have no doubt that you will be getting lots of good sponge information on that cruise. Oh, there's also sticking its head out of the rock just underneath this eel is a different kind of fish. So just pause on the coral and let's check that fish that's coming out. Wonder if it's an eel pout or something. Thank you. It just tucked his head back down. What are they? There's eel pouts or ophidiids. He's shy. Or he's wise. Stuck his head back down. Okay, he went back into the crack in the rock, so.
I'd go with wise. He doesn't want to be slurped up.
Sure. It had a very bulbous head and then a very long fin in the backside of the body. All right, so what's this white coral? I'm thinking it's either a bamboo coral or it's another one of the anthithelids. The anthithelids are always a little bit tricky when I see them from a distance. Yeah, that's another antithelid. Okay, that's the one we were imaging earlier, and I think it's the same genus. I'm not going to say same species, but that we collected on Dive 1. These are what are called Sclerexsonians, and many of them have an axial skeleton that's more like concrete in that they've put together lots of different sclerites. So instead of a proteinaceous skeleton that when you strip off the tissue, the skeleton remains behind, this would be more like a dried sand that you could rub with your fingers and it would just sort of come apart. Okay, thank you. Whenever video's happy, I'm happy.
By proteinase, you mean protein-based?
Yeah, thank you. A protein-based skeleton. And so, you know, I think it looks like you're looking at a woody skeleton if you look internally, but instead of the cell walls, it's made of protein. There's another Acanthogorgia. This is what James was referring to earlier. You can see it's got these very thin, tall polyps that stand off of the skeleton, and they really don't have any tissue to retract into. So that's one of the ways that we can relatively easily recognize Acanthogorgia from a distance. And 2 cup corals. They're pretty good. Actually, multiple cup corals down there. More sponges on the side of the rock. I think another actually that might be a madrepora, the colony that's on the left. Another stony coral. So again, all kinds of stuff growing here. Yes. It's a good question. Are you on the Okeanos line? Yeah, I just want to make sure people could hear your question. It makes it more flexible. Sorry, let me get to your question in a second. I noted earlier when we first got down, I saw an Oreo, and there's one of those Oreo fish in the background, just making a note of it again. So I don't know. Okay, thank you. So Arvind asked the question if there an energy-based advantage to having a protein skeleton versus a calcium carbonate-based skeleton. I don't really know the answer, but what I can say, I'm looking at things trying to name them, so I said name. What I can say is that the protein-based skeleton tends to be more flexible. The calcium carbonate tends to be more rigid, so it may have originally been some adaptation to living in a high current flow. And one of the things you have to sort out is what is an evolutionary response right now versus what is something that you have inherited from your ancestors. And so why they may have them now versus how they originally evolved them, I'm not sure. One of the things we can clearly see in front of us is that in this habitat, both the protein-based and the calcium carbonate skeletons are perfectly fine because we see them growing right next to one another. As we come into this sponge, I'll note the bottle brush looking thing on the upper left is a Chrysogorgia, so that'll be our first of the day.
Hey, buddy.
And then right behind that feather looking thing is a black coral Parentipathy. And then there's a cup coral in the back. Yeah, so here's this sponge that Joanna was pointing us to, and I have to scroll back up. I believe she said Pheronema. And there's all kinds of comments in the chat room. I have to start reading some of what's being said, but it looks like there's some kind of associates, maybe amphipods on the inside of this sponge. And this is a glass sponge, hexactinellid. I see a small crinoid living on the outside. Let's see. We'll peer down in here. Good little view. Oh, thank you. There we go. Huh.
What are you looking at?
Well, I'm looking at all these curled up pinkish things that I've assumed were amphipods at first, but now that we're zoomed in a bit, I mean, that's an amphipod. The lowest one is definitely an amphipod, but these other ones, I'm not 100% certain they're all amphipods, especially this largest one that's second from the bottom. I'll have to wait for what my colleagues have to say. Thank you. And you can get a really, really nice view of the complexity of the siliceous skeleton, the silica-based skeleton that this sponge is laid down. And so these are structures that are called spicules, and the tissue is kind of draped on top of that. There's specialized cells that secrete this. Really nice. And then water flows through that system, and the cells that are in there have collar-like structures that they can pick up bacteria and so on. There's some possibility that they also can feed on dissolved organic carbon. Thank you, Pilot.
And it looks like the exterior has some vertical spiraling in the color. Is that, do you see what I'm looking at? The banding?
Yeah, I'm not sure if that, to me, looks like there's probably some high point in the skeleton that's capturing more sediment.
Okay.
Clear. Let's move on. Why don't you direct some interesting things while I check out all these comments in the chat room?
All right, so we're seeing a fairly heavily manganese encrusted surface. We see this strange little, let's call it a patio. It's a nice little shelf. Yeah, very, very kind of semicircular. And then above that, you actually see all these pillow lavas that almost form these tubes. So, these pillow lavas as they're erupted, they form these large tube-like systems that can kind of fall down from their high point of where they erupted. So this might be actually a little, if it's still in place and hasn't been faulted, or its original orientation hasn't been too disturbed, given it is probably about 1.4 million years old, it might have been flowing from that little peak at the top, so that might have been the vent of a little pillow vent. And as those pillows were erupted out, you can think of kind of squeezing toothpaste from a toothpaste tube, and it kind of just falls down this slope, and you get these large tube systems. That kind of little platform on the bottom of it might be an underlying sheet flow. So a lot of times the initial portion of these underwater pillow eruptions are very voluminous, and the more volume output that they are over a short amount of time, the eruptive rate, they tend to form these large sheet flows. So these large, not necessarily thin layer, but flattish, almost very fluid flows, opposed to these pillowy structures, which are more common when the eruptive rate is lower.
So it's got a good view on stream 3 from the blue view sonar.
Yeah, so one of the other things we're looking at is this sonar that's forward-looking on the ROV. We call it the blue view, and that gives us an idea of whether we have any of these similar features, of these hard, prone returns beyond our visual range of sight. But that's some beautiful pillow tubes, and we see this little thing swimming by. It's about my scientific expertise on biological specimen identification. Now you missed it, Scott.
Oh, it must have been something really spectacular.
Yeah.
There's some very interesting sponges here, though, and yeah, let's just kind of zoom in and work our way to the top of this little pile. So what is this, a little...
This looks like a little eruptive vent.
Yeah, cool.
That fed some of these pillow tubes.
Nice. Isn't it bizarre? This is what I thought we'd be doing yesterday.
I know. I
just
give up. You know, I went to the youngest, biggest volcano I could find and saw nothing, and then we go to something 1.5 million years old.
I think you did ask me to pick a biology dive that had lots of geology, so maybe today is the day. Nice sponge that we're moving into. I will rely on Joanna and Manuela to give us some ideas of that. I still haven't finished going back through all the chat comments. This is really exciting. You know, this is a Sunday, and we've got a full chat room of scientists who are all really eager for this opportunity to visit the deep sea floor. Lower right, I believe, is a coral we haven't yet looked at today. You watch. We're going to go in, and it's going to be another antithelid, but it just has that purple tint to it, and I neglected to get a close look at the bright purple one that we saw at the very beginning of the dive. Yeah, I can see that this is going to be an Antithelidae again. Not sure what the little coiled thing is at the base of it, but we'll see when we zoom in here. The sponge, let's see. Oh, I see. It's just a holdfast. It's the base of the sponge. Yeah, this one has like a purple tint to it. This may be a plexaurid. Kind of interesting. Yeah. So I've seen this before on some of the genus Paramaricia, where the sclerites are a different color from the soft tissue, and so you can get a colony that looks to be two colors. And so here it looks like we have yellow polyps in a purple matrix. And yeah, if we could keep going up like the way you were there, pilot, I think higher in this colony there may be a rather large aplecophoran worm. I'm not sure. There was something. I don't think it was the arm of a brittle star. Yeah, right there. What's going on there? Yeah, I'm pretty sure that that's a specialized predator of octocorals. It belongs to the mollusca, and there's some systematic reorganization going on. I usually call them aplecopherans, but I think now this one, I don't remember if it's a selenogaster or Caudofoveata, but All big Latin names. Bottom line, worm-like mollusk specialized for feeding on corals. Okay, thank you. So Joanna, you're pointing out a sponge. Can we zoom in? Is it stocked? Are you talking about the one that's to the left, that large white one, which is what we were coming in for originally? But yeah, if we can get a look at that sponge, and you notice we want to see how it's attached to the rock. So the one just to the upper right of what we were zoomed in on the next rock. Photobomb by Honorio. Kind of gnarly faces, gnarly knobby faces, big eyes. Maybe Ken can tell us a little bit about that dorsal fin. I'm not sure what that little pointy dorsal fin is all about. No, the rock behind and to the left. Yeah, it's a really large white thing in the center. Yep. Thank you. Yeah, so one of the ways I know that the sponge biologists use to do their identification is to look at how the sponge is attached to the substrate, and that can be informative. And you can see sticking out of the base there these needle-like structures, so those would again be some of the siliceous fibers, the silica-based fibers that the sponge produces. This is again a glass sponge, a different type than we looked at earlier. They can be really mesmerizing to look at if you start to get into the complexity of the architecture. Nice view. Those little crinoid, or at least the cirri, the feet of a crinoid, if you will. I don't actually see the arms. That's kind of interesting. Maybe it's just very young and it's growing. Yeah, there in the upper right, that's more what I would expect to see. And these just look like the grasping feet-like cirri. Interesting. That's another echinoderm related to a sea star. But they perch and their mouth is oriented away from the bottom, and they use their arms to capture things out of the water column. Little hermit crab up on the top lip of the sponge. Don't know where he found that snail shell. The snails are few and far between down here in the deep. But it managed to. All right, that's a fantastic view. When video's good, I'm good. Our colleague Isako Matsumoto from Japan has pointed out that the octocoral genus Swiftia also have a difference in polyp color versus scleric color, and so you see these multicolored colonies. There's another eel flying up above the rocks, and then this sponge that's encrusting the rock there in the center is kind of interesting because I'm not sure if parts of it are alive or parts of it are dead. You know, there's some of it that's yellow and some of it's white, or if it's one sponge on top of another. In the background there, that white tall colony is almost surely an unbranched bamboo coral. What do you think this is, Ashton, this white that's in the crook of this pillow? Is that geological or is that biological?
I think probably maybe cemented together pteropod shells and some biogenic oozes.
Pretty clear to see all these pillows. So these would have been extruded from up, or is this what you were telling me the other day that this is in place?
I think this probably would have been extruded from the top of this almost cone-like feature, and it, as it came up, it flowed downward, forming these large kind of elongated pillows. Pillows is the terminology we use. It's not necessarily very descriptive because pillows implies that they're kind of these isolated little packets, and really they're kind of these big, long, stretched out pillows. Almost think of less of a pillow and more of a body pillow that kind of stretches for feet and feet and feet. they don't really form those isolated individual pillow structures unless they kind of detach over steep topography and fall off. I did not bring my body pillow on this trip.
Wow. Let's have a look at that large orange colony back there. I suspect that's a large Leopathes, but
and it's interesting because it seems to be growing at the very top of this little volcanic cone.
Yeah. And so we talked a little bit about this yesterday, that, you know, all of these sessile organisms, and sessile simply means that the animal has attached itself to a substrate. They're suspension feeders, meaning they're getting their food out of the water column. And so you want to be in a place where the water is moving fast enough to deliver enough food per unit time. And that tends to be on these features that stick out of a flat bottom. If it's really flat, then the current sort of can dissipate quickly. But if you're on a rugged bottom, then you get a lot of turbulence and eddies, and that can keep some of these food particles in place, as well as ensuring that you're out of the static flow right at the bottom, or I don't think there's such a thing as a static flow.
No, I mean, from my understanding, you know, for some of the larger seamount features, you know, some of the fastest current is actually right at the base of it, depending on the flow direction, because you get this kind of this basal current that goes around the seamount feature, so you actually get kind of erosion of the sediment around it.
Right. I'm referring more to being in contact with the bottom. Yeah, so this is another Leopathes, quite a large one. You can see the lasers that were on there for a moment. So this is probably a very old colony. And if you do continue to swing around here as you are, let's continue to swing around all the way to the white colony if you have enough tether. There's several of these colonies here. You can see there's a real difference on the two faces. So this, I guess, would be the upslope face that we're coming into. There's more colonies growing on this side, which would suggest that the currents are better on this side. Oh, there's a... Is that a salp chain, I think? Or is it a siphonophore? Oh, it is a siphonophore. Yeah, good call. This is a colonial hydrozoan that's now photobombing. You can see the far end of it. You could see the pulsation. Those are the swimming nectophores. There's a little float at the top there.
Reminds me of a maple tree.
Smaller leaves, maybe? Yeah, beautiful view. I really need to pull out my note here to tell you something about the rate of growth of this thing. You'll be amazed, I'm sure. So let's see. My notes say that these studies show corals grow very slowly, with some growing more slowly than others. The slowest growing, so far as we know, are Leopathes, which is this orange colony we're looking at right now, which increase in thickness, so the radial growth, by less than 25 microns per year. And that means it would take more than 50 years to grow 1 millimeter thick. It's incredible. And somewhere on here I remember putting in, yeah, a human hair is 75 microns on average, about 17 to 180 microns. And so we're saying that in a year, this grows in thickness less than the width of a human hair. So as you can imagine, such a slow growth, there's a lot of concern about protecting corals like this, because if they are damaged, it's going to be some thousands of years before we see scenes like this again, and that are providing the substrate for all these other organisms in the community. Yes. Yeah, thank you. We don't need to go back in on that orange colony, but let's check out the white whip-like one in the distance and the white fan ones in the foreground. Thank you.
And so one of the ways we can tell whether this was actually a cone that looked like it was erupted in place is when we come around onto this other side, we can look for similar pillows that we saw on the front side and see if we also see the same similar pillows on the back side. And that means that as those pillows erupted out of the top of it, they flew kind of radially down in all directions. If we look on the back side and it looks like it's more rubbly, then that means this whole probably system was probably faulted and it's not necessarily in place.
Looks like there's a crab on this bamboo coral, so this almost certainly is a bamboo coral. It's not the whip kind, because you can see one branch coming out towards us right around where that squat lobster is. Several cup corals here. You know, this black stuff, I'm not sure if that's what they call a tar sponge or if that's geological. I'd love to have your input on that.
Yeah, I don't think it's necessarily, the only thing I would imagine would be a manganese coating, but I would expect that to be much more uniform and less dark.
Okay. Pilot, when you are capable, if you can go just a little bit lower on this colony, I thought I saw streaming out from here some very thin, yeah, there we go. See those really thin, delicate lines? They're going off to the left in the current from that colony. I'm not sure if that's mucus or if there's a bunch of really small ctenophores living on this colony. That's interesting. It's not mucus. So another possibility is that there's a the name for some sweeper tentacles. There's something called a sweeper tentacle that some corals are capable of releasing, and so maybe that's what that is. Usually it's when they're being disturbed by something. Let's also take a look at all the dots that are on the rock here. It's interesting. Yeah, so I'm looking at all this stuff that's coating the rock. You can see there's a lot of circles back there that seem to be growing as a mat. And what I'm trying to determine, those are probably zoanthids. Pilot, if you're able to turn to the left, you see, looking at the back side rock, what's coating the rock. Yeah, those are definitely zoanthids. And Pilot, just for awareness, following getting a view of that, there's been a request to make a collection on the top of this rock feature. Correct. So I'm not sure if we're doing something for video or for me right now. Oh, if you go to the left, there's a wide open view of it. Get away from this coral there. There you go. The face of the rock to the upper left. I realize it's sort of tucked under there, but I think a zoom is fine. In fact, I already know that there's zoanthids. I saw that on the view that we just had. So the zoanthids are another kind of hexacoral. They're related to sea anemones, but they're often colonial, and so I think that's what I'm seeing here. All of those little individual polyps are connected by a sheet-like layer of tissue, and they're just spreading across the rock. All right, so we're going to get into the place to collect this whip coral. We've had a request from our colleagues on shore saying that, as far as I know, this is common in the area, but it has not been identified. And so again, we get back to this issue of, we have this vulnerable marine ecosystem. There's a swimming crinoid, by the way, just to show you that those feather stars are capable of swimming when they're disturbed. Although, based on data that we have from long-term camera deployments, they tend to prefer to sit in the same place for a year or more on end. And I think I see in the very top of the screen one of our Hyperion amphipod friends pushing its salp house. So, we're going to get in position to collect this bamboo coral. So the point was, vulnerable marine ecosystem, we'd like to know what are some of the components of it. We can see it, but we haven't been able to identify these things yet. And so we'll get a piece of that. I'm also seeing in the center there, to the right of that white fan, there's a Parentipathy's black coral. So that's the first time I've noted that name today. Copy that, thank you. For more information on this coral, I suspect it would be easy to cut or to bend snap, so whatever's easiest for you. And I have to look at the size a little bit more. Ideally, I would love to get it just below the branch point, but I'm having a feeling that might be too long, so we'll just, let's say, go for the upper 7 inches or so. Thank you. The lasers just passed over that coral, and from that upper crook where it bends to the left to the branch that's coming out to the right, that was about 10 centimeters across. So I would guess that that branchlet that's going up is probably at least 20 centimeters. So one of the challenges about a collection like this is that, there's nowhere for the ROV to settle. So we've got to try to position it to be as stationary as possible before they pull out the arm and try to clip it.
Now, so some of those black patches on the right boulders, I think that's just probably less manganese precipitation on those. So whether those have been just kind of scraped or more freshly broken off surfaces.
Right. So there was a coating deposited and now it's been removed.
Or it was more intact and then broke off later, so it's had less time for that coating to be deposited.
Right. Oh, I think I see a rock pen over here on the right side, just to the left of the ophiuroid. I'm going to make a note of that in the annotations. That looks like maybe a codling. If it's not too long, yeah, but that's the question. We'll see once the claw gets near there. let's call it Keratoisididae. Well, we got one arm of the squat lobster that we can use for DNA. I mean, no, seriously, that's DNA-able. So that's a beautiful clipping. One of the reasons that we ask for the branches is that it's taxonomically important for identification about where the branches arise. Excellent. That is in starboard outboard biobox. Another successful collection. You are absolutely correct. It's my old right-left problem. Thank you. So, unfortunately for our data manager, both of our port samples today I've labeled as starboard. I've got it right on this form, but not on the Excel form. I'm pretty sure that in ROV sample collection, both samples that went into port are shown in here as in starboard, but they're correct on here. Can you also make a note for this one that I think I saw that chirostylid leg go in there as well? I want to get that. Thank you. Yeah, I think we're ready to explore a little bit more. I think we could probably spend the whole day on this one little cone, but thank you. So we've now made a collection of a black coral, that bamboo coral, and Joanna has asked for if we see another of those Farinema sponges to collect, and she pointed out that that sponge is very mucousy, and I know that that black coral is very mucousy, and I know that bamboo corals are very mucousy, so I don't know what you all are trying to do to me tonight when I'm working in the wet lab, but there's going to be a lot of mucus in there. What's, I guess these are just scattered rocks, we're just high. Pilot, if possible, on the left side center screen, that wiry looking thing that's growing up. Thank you. I suspect that this is a bamboo coral, and I will go out on a limb and suggest that it might even be genus Ecnomyces. Kind of has that sort of loose, three-dimensional growth form. Copy that. Why don't we go ahead and zoom in on the fish? Can you just do that? I think that, thanks. Sorry, not that fish. Top of the screen. We know that fish. It probably went off the top. Yeah, right, that one. That's the one. Excellent. Is this a codling, maybe? Yeah, I'm pretty sure this is a morid of some kind, codling. Yeah, no, that's not necessary to chase. That's good. That's a good view. I just wanted to, since we were sitting there. Thank you. Good stuff. Let's see, anything in the foreground as we wait for Sirius to get ahead a little bit here.
And so if you look at some of these rubbly blocks, you can see like the one kind of almost center screen right now with a little white dot on it.
This one?
Yep, that one. See that outer surface is kind of this brownish muddly, but the fresh bottom surface, the fractured surface, is darker.
Right.
And you can see that in some of the rubble blocks around here. So that gives you a good idea of, this was rubble that was broken up later, probably at a much later date.
Got it.
And to give you some idea of where we are, if you're just joining us, we're on the Mid-Atlantic Ridge. We're on the southwestern-ish face of a ridge that's about 1.4 million years old. It's in a leftward step-over of the north-south trending Mid-Atlantic Ridge, a very small transform fault. It's just above that transform fault on the eastern segment of the ridge. We're about 340 kilometers from the Azores, 340 kilometers from the island of Corvo, the northwest island in the Azores. We're at a depth of 1,060 meters below sea level, and we'll be coming up to a minimum depth of about 980 meters below sea level. We're currently at 42.339 degrees north latitude and 29.148 degrees west longitude, and it is currently 13:11 UTC on July 24th.
So folks on shore, just for some context, remember, the main camera that you're watching is the Deep Discoverer ROV. It's on a tether that's about 30 meters long, so think of that as your dog on your flexi lead. And then it's attached to the Sirius, and the Sirius is attached to the ship by the winch cable. And so the D2 camera basically can mobilize within a 30-meter diameter ring. But if we want to go further ahead, we have to wait for the ship to move Sirius, and that's basically what's going on now. There's a lot of things that we want to zoom on, but we're waiting for the ship to allow us to go forward. And right now, pilot and navigator are conferring, and when they're finished, we'll be able to ask for zooms. Yeah, I did video. Thank you. I'm interested in that. Oops. Excuse me, pilot. I thought Nav was talking to you, and now I understand or not. What we're interested in is actually everything over to the right. Now that you've drifted over here, my apologies. Yeah, exactly. Yeah, good view. Nice urchin over there. Yeah, so there were several interesting sponges along that rock, and as we're moving from this pile across the low point to the next pile, within the low point, there were two slender whip-like branches sticking up at the bottom. So if we could just look at those as we go by, I'll try to be... Thank you. Another one of those brachyurine crabs going by. Oh, actually, you know, our initial target was that wiry bamboo, and we still haven't had a close-up of that. So I kind of want to start at that one and move right. But Roland, I understand the two whips you were pointing out. I still haven't seen them. That's Parentipathy's black coral with one or two squat lobsters on it. All right. Yeah, let's go in on this one. So the request from shore, one of the reasons we're looking closely at this bamboo coral is just to ascertain whether it's the same or different from the one that we just collected. And one of the challenges for identifying all of these species that we're seeing from in situ video is that we're taking one snapshot in time. And of course these corals are growing for decades and hundreds of years, and so they're changing their morphology. So it may have been that the one that we sampled earlier was just a younger stage of this one. And so getting a good look at it and then comparing with what we have in the lab later on, we'll get an understanding of how they grow. You know, yesterday you asked me about that sponge on the stalk. You know, does it begin to have its thick body when it's small or when it gets big? And this is sort of the analogous question. So, pilot, when you're ready, what I would like is a close-up of the polyps. I'm still kind of holding to my initial guess that this could be Ecnomyces, the genus Ecnomyces. Almost assuredly, it would be in our genetic clade D. Yeah, nice. So you can see the polyps that are closed up have kind of a little volcano shape, kind of good theme. And there we can get a great view of the sclerites, these calcium carbonate secretions that the coral makes. And the tentacles have been withdrawn. And so you can really see with the ones that we call the intertentacular tentacles that are sticking up above the main body of the polyp. But otherwise, we can see from this view that all the tissue is really well endowed with these sclerites. The polyp body wall, up around the margin where the tentacles are, and the tissue between all these polyps. And then we also saw there how dense the polyps were. That was a terrific view. Thank you. We can now move back towards the right and look at some of these sponges. So you can see the nodes there, and the tissue is so thick that I'm not 100% sure that I can say that that's an internodal or nodal brancher, but I think the branch is coming just above or just distal to the node. So that would be an internodal brancher, which would make it support again that this would be Ecnomyces. It's a massive. So I've now seen 3 positions where I'm pretty confident that it's internodally branching and coming just after the node. So that supports that this is Ecnomyces. There was some suggestion maybe it's in the J clade, is it Keratoisis? I think we got a pretty good ID here. Pretty fearsome looking sclerites there, which are presumably defensive. And that's, I see these things a lot, and I imagine if Ophiuroids, brittle stars molted, that's what it would look like. Mm-hmm. They don't. So I'm not exactly sure if that's just a really young one or what. All right, thank you for that. Yeah, so if we could have a look at this big frilly sponge that's just come into view, and then after that, the tube-shaped one after it, and then after that, the yellow one above it. All right, Joanna, you're on. So, I wonder if this is the one that has all the zoanthids. Yeah. Is this, the name Aphrocalistes is coming into my brain. We'll see if that's correct. But in the Pacific, and I think in the Western Atlantic, we see these sponges, and they're covered by zoanthids, and I think that's what I'm seeing here. You see there's a whole bunch of like tentacles, and there's all these dots on the sponge. I believe that is a symbiosis between this colonial anemone and the sponge, and we see it repeated in many places. So Joanna says it's not Aphrocalistes, so I guessed that wrong.
It's okay. I still believe in you.
Thank you.
You're welcome.
I think everything else I said is accurate. Just the name I can't recall. Beautiful. Again, this silica-based skeleton. It's interesting to me, and I think about sponges, and I look at sponges. I think they're a lot like colonial corals in that there's a lot of different ways that skeleton can be laid down, and so the morphology changes quite a bit. Actually, we should look at that one, the yellow one, at the lower right of this white sponge that we're looking at. It's just tucked underneath it there. Very good. So Joanna is suggesting Conolasma as a genus. So Joanna, we're going to zoom in on the yellow one, and maybe we can also see the wall of this white one as we zoom in. Maybe it'll take care of both requests. And certainly we have good video of it that we can review later. Yeah, so this looks like it's built as a series of tubes that come together in a ball. And I don't know if this is a demo sponge or a glass sponge. I wonder, is that a Phareid in the family Phareidae? I'm just throwing out names now and seeing what comes back. Gorgeous. So I think this one is growing from the left side there off the wall.
Yeah, in the upper left-hand portion of the screen, that really white portion of this, is that the stalk of it?
Yeah.
Or is that separate?
That's a good question. It does look spongy to me. So I think you're right. I think that might be the stalk of this yellow one.
All right, I'm done. I retire.
Okay. So yeah, if we can pan up just a little bit more on the white one, we'll get one final look at that wall. That's good.
Joanna says the yellow one is probably Hertwia fossifera.
Great.
I just wanted to pronounce that.
Yeah, that's nice. You did a good job. You can see all the zoanthids or anemones. Thank you. So we can move on to the next sponge on the wall. Thank you. Great stuff. Yeah, so this looks like a euplectellid of some kind, a vase sponge, and we were seeing these in the last couple of dives. We were talking about that sort of sieve plate-like structure on the top of the tube.
You can see just at least this portion of this outcrop that all these sponges are attached to, just how much more rubbly it looks, right? Yeah. It looks like just a concretion, a kind of a cementation of a bunch of little individual pieces of,
exactly.
rubble.
And is that what it is?
Yeah, that's what I think it is. Something that's been, we're at this place where we're kind of on the southwestern face of this normal fault, so it's been uplifted. Everything's kind of been disturbed. It's created a bunch of failure features, a bunch of talus, a bunch of rubble. It's sat here for a million years, and it's kind of just been cemented together with biogenic ooze and
But does that mean it's broken again? Because now it seems to be uplifted.
Yeah.
Okay. I think we're going to get a good view of that sieve plate on the top of this one because we're at a really good angle.
That one looks kind of like the one we just looked at. Same with on top right.
Yeah.
Yep.
I agree. All right. Great view. So this is yet another glass sponge. You can see all these silica spicules that are extending from the main axis of that sponge. And then this, what I've been referring to as a sieve plate covering the top. We had a really interesting discussion about this yesterday, that it's not particularly serving the flow needs of the sponge. That would be likely the X current opening. But it may provide protection for associated organisms that live inside the sponge, particularly shrimp. There does seem to be a pair of shrimp that are adapted to live in these sponges, and there was even a suggestion to some data that suggests the shrimp are benefiting the sponge by their excretion products providing nutrition to the sponge. Oh, there we go. We see one crawling inside. Thank you very much. So there's one of those shrimp. Might be coming up to check out what's this light outside my front door. And typically we have a male and a female, and I'm trying to see if I can see this one carrying eggs underneath her abdomen. What a fantastic view. Look at that.
Help me!
No, this is the home. This is it right where it wants to be. In fact, it enters as a larva, so it's small enough to move through the holes, and then it molts into this larger stage. At least that's the way we all interpreted it until Ashton had a look.
Well, I don't think it's a conscious decision to enter as a larva into this system.
I beg to differ. I think the larvae have a lot of control about where they go.
You think so?
And, yep. And so probably can chemically detect this sponge and then, choose whether to enter it or not. And that's the story I'm sticking with. Wow, what a spectacular view. That was really nice.
Yeah, great example of what we talked about yesterday.
Yeah. So presumably there's another one inside there. So, that shrimp is pretty well protected from these various fish that are swimming around looking for a tasty bite. It's interesting to think of all of the silica that the sponge has to take out of the water in its dissolved form and then deposit in this skeleton that is growing. I'm not sure what the point of all those silica fibers sticking out is. I wonder if it creates some turbulence and allows for the currents to sort of swirl around and then can be more easily taken up by the sponge, perhaps.
Could they be, sensory from the standpoint of making sure that it's staying within, kind of away from the surrounding rock face or anything like that? Can it help guide growth direction?
So there are some
video,
limited
yes.
sensory capabilities in some of these sponges. They don't have a nervous system the way we would think of a nervous system. But they are able, I know, at least some of them, to react to compounds in the water and so stop the flow of water, for example. But, yeah, I'm not sure about that as an explanation. Joanna offers a possible idea of either Euplectella or Rigadrella for that sponge. Absolutely beautiful. Another one of the Hartwigia down there. All right, so let's move on a bit. I think we've moved, 5 meters?
In the last 3 hours, yeah.
Exactly. I mean, it's stunning. There's all kinds of stuff here. We have lots of scientists on shore who are really excited about what they're seeing. First opportunities to sample some of these for ID. It's great stuff. We never have enough time to do everything that we want.
And so you were asking about those kind of whitish gray, brownish patches on some of those rock outcrops. To me, it just looks like the same kind of pteropod shells that we've been seeing, but maybe just have sat there longer and kind of concreted together.
Yeah, so they're sort of cemented together the same way you were describing those cobbles.
Yeah.
But still, with Joanna here, earlier I saw some really glossy, I guess might be the word, black patches on the rock, and I wondered if they could be something like a tar sponge or if it was some encrustation, or I don't think it's what you're describing about, the newly exposed surface. Those look really different.
Yeah, those look different, and the other ones that you pointed out originally were much more patchwork.
Yeah.
Yeah, just not as you'd expect in kind of places where it had recently failed.
Yeah.
We could always scratch one.
This might be a Geodia sponge that's coming up. I think the sticks might be spines like of this urchin, so maybe we can have a pass by this urchin that's just going out of frame on the upper right. We get Joanna to tell us what she... Oh, I recognize this. Oh, this is... You know, I thought I made some notes about this the other day. Farinema, thank you. Actually, if we could zoom on that fish that's swimming just above the sponge, that would be really helpful. Really small. Yeah, it might even have a parasite on its side. Or it was just, no, it does seem, I don't know if that was 2 parasites or it's just swollen in the middle of the body. Oh, it's swollen. It's got a fat tummy. Must have just eaten. Very small little eel. So actually this rubble, this is a lot of coral skeleton rubble. Thank you. Good view of that eel. I'm noticing the tail has a different structure than the ones we ID'd earlier, so I bet we're going to find out from Ken Sulak that that's a different species. So yeah, this is all coral rubble, not pteropod shells. Probably cup corals and Lophelia and Madrepora. Bird's nest sponge. Thank you, Joanna. That's what I was trying to remember. The Farinema carpenteri. So I think this one's pretty common out here. Little crab tucking itself in a coral rubble down there. You can see how loose that rubble is as the ROV toe there on the right sort of pushes into it. So you can see why it's called the bird's nest sponge. All these silica fibers at the base that it uses to attach to the substrate. And then as we come up and you see the more white part of the sponge, that's the living tissue. And again, what you have to picture is that on the inside of the walls here, there's this massive plumbing system. There's all these very small openings to that plumbing system on the outside of the sponge. And the cells in the interior have flagella that they beat to create fluid flow, and they push it towards that center osculum or that center chimney, and that creates negative pressure or suction that pulls water in from the outside, and that's basically the way they feed. There's a little octocoral growing on the top of this sponge, several crinoids, the feather stars with their arms curled up. And a bunch of other even smaller stuff. Looks like maybe a little amphipod there in the center. Maybe some kind of, maybe it's a platyhelminth worm or something that's just to the right of those octocorals and above that small brittle star. See, it's kind of looks like a slug the way it's moving around. Very nice. So let me consult with Joanna. Pilot, are we in a position to sample, or have you already put in your move? That sponge we were just looking at. Thank you. Yeah, Joanna, if you're listening, I put a follow-up question in the chat for you. Get some advice on how to sample this. It might be so small that we can only take the whole thing. Yeah. Okay. Yeah, so the information I'm getting from shore is that this is very fibrous, and it would be very difficult for us just to cut out a piece of it. And so given its size, we suggest just picking up the whole sponge. Thank you. You can see there that fibrous nature that Joanna was talking about. Might have got some coral rubble as well. Oreo's like, what's going on? On shore, if you go over to camera 3, you'll get a better sense of what's going on with the sampling here. And there it goes into the port side bile box. A starboard. I did it this time. I'm reversing it. Jeez. Starboard inner.
Yeah. Mic check. And what we see in the background is more of these skeletons of the corals.
Thank you, pilot. Very nicely done. So we've got an intact sample of that Pharonema glass sponge, and it had a variety of associates with it, corals, crinoid, and who knows what all. So there should probably be a lot of processing of that later on. And we'll preserve some of that sponge tissue for morphological analysis, looking at the tissue, looking at especially the spicules, the shape, diversity, and abundance of which are important for taxonomy. And then we'll dry a large part of it after we've picked off all the other organisms. So this might be a good time just to remind everybody or introduce ourselves for the folks who have just tuned into the live stream. You're watching live video from the Voyage to the Ridge expedition on board the NOAA ship Okeanos Explorer. My name is Scott France. I'm a deep sea biologist at the University of Louisiana at Lafayette and your biology lead. And I'm here with my geology lead.
Who's that?
That's you.
Oh, okay.
Yeah.
Yeah, my name is Ashton Flinders. I am a research geophysicist, mainly studying earthquakes and variations in the gravitational field of Hawaiian volcanoes at the Hawaiian Volcano Observatory in Hilo, Hawaii, with the U.S. Geological Survey.
Awesome. Front row, if is it appropriate, would you like to introduce yourselves, or is this not the right time?
Sitting in the front row, operating the ROVs. At the far left chair, we have navigator Lars Murphy. Sitting pilot Chris Wright. And I'm Levi Unima, sitting co-pilot. And in the far right, sitting in the video chair, we have Roland Bryan. And we have Anna Clipping in the back row. And we are all with the Global Foundation for Ocean Exploration.
Awesome. And also in the back row, we have our data manager. Excuse me. We have our data manager. Yeah, go ahead, Arvin. Introduce yourself.
Hi, everyone. My name is Arvin Chandran. I'm a data manager and research associate with NCEI and the Northern Gulf Institute.
Whole lot more sponges. Oh, wait a minute. Was that a sponge? Yeah, okay, that was a sponge. So a whole lot of sponges on the rock, crab tucked in there. Joanna, I don't know if you heard this earlier, but one of the rocks that we collected yesterday, I did manage to scrape off one of these encrusting round sponges, so we do have a piece of that. There's a stony coral here coming in the top center, and all the frilly stuff that you see growing off it is hydrozoan, what we call hydroid colonies, another colonial kind of anemone. And then the bright white one that's facing off the brown rock, that's a hydrocoral, probably the genus Crypthelia. And then the Leopathes black coral is the large orange thing that you see in the back. We've seen several of those colonies and managed to collect a piece of one of them earlier. There was a lot of interest in that in determining how much of that Leopathes black coral might be up here. More of the Sclerexsonian colonies. You see cup corals. Those are the individual little white calyces or cups that you see emerging from the rock. Let's see, what else do we have here? If we could zoom just the top, sorry. Every time you move, I go to the opposite direction, right? Yeah, top center. I'm not sure what that sort of pinkish fan-like thing is in the center. I think it's some kind of black coral, but I don't recognize it. Huh. I guess, my best guess is that's the genus Parentipathy. It's 100% it's a black coral. Its coloration is a little bit more pale than I'm used to seeing with Parentipathy. Parentipathy is a black coral that grows like a fan, has branches, and then sub-branches on the branches, and then sub-branches on those branches, and that's kind of what I'm looking at. So that's my best guess at that. Thank you. Oh, Les Watling noted that I guess we passed over a bramble bamboo. I missed that one. And that's one that kind of grows as a little bit of a mess, very thin skeletons, and looks like somebody has played with a ball of string and then dropped it. That's the way I would describe bramble bamboos. Yes, we are. Thank you. Seeing several of these small midwater fish reflect in the water column, probably cyclothony that are coming down close to the surface of this feature.
Now I'm going to ask you a question, and I don't want it to be incendiary.
Oh,
but
it's going to be good, I can tell.
But I do want it to be maybe some basic Bio 100.
Okay, stand by. Pilot, can we get a zoom on that star-like thing in the top center? Thank you. Go ahead, Ashton.
We're getting a lot of taxonomy thrown at us. Why should I care? In terms of, like, what is the importance in terms of finding out the relationship of these organisms that we're looking at?
Yeah, I think that's a very fair question. And so we're trying to, in a sense, catalog diversity. And so having names for everything allows us to sort them out. And all of that Latin terminology that you hear is basically a formal way for people who are in this business to all know what each other is talking about. Everybody who has a certain career knows that they have a certain vocabulary associated with their job. And it means you don't have to explain yourself every time you talk to a colleague about what you're looking at. So we'll come back to that in just a moment. So this is a sea urchin, another echinoderm, and it's I'm sure this is a sideroid of some kind, and I say that only not because I'm an expert, but because Chris Ma identified something similar to it a couple of dives ago. And yeah, Les Watling is backing that up. And that's partly based on how thick those spines are. Thank you for that view. Yes, we are. Thank you. So the other thing we're going, Ashton, to follow up on your question, we're going through a bit of a taxonomic and systematic revolution. Actually, sorry, just above the lasers now. You see that? Yeah. I thought at first it was a sea pen, but now I'm wondering if it's another Okeanos bamboo coral. We've been seeing plenty of them today. They're the ones that I described earlier, can grow on a rock or in the sediment. I suspect this one's growing off of the... yeah, that's exactly what it is. And it's growing off of that rubble. And just like the one we saw earlier, you see it's got its little squat lobster in there. Thank you. Good view. Good move on. So systematics is undergoing something of a revolution with the advances in molecular biotechnology and gene sequencing. And so one of the reasons why you may hear we say, well, that used to be called this and now it's called this, and we're not sure if that belongs to this or that, is because we're trying to align the names and the groups that these things belong to their evolutionary history, which is what we learn from the DNA. And so that also gives us a shortcut to understand, well, how have these things evolved through time, and how have they adapted to their environment, and are certain characteristics possible to evolve multiple times, or did they only evolve once?
Mm-hmm.
And a good example of that is the rock pen that we saw the other day.
Yeah,
As I said, that appears to have evolved once. There's only one limited group so far that has figured out how to live on a rock.
And then what was the organism we saw that you had talked about had come from the rock and then had evolved to be in the sediment and then gone back to the rock?
Yeah, so that was the sea pen, the rock pens. And so the sea pens are octocorals, like the bamboo coral we just looked at. That bamboo coral was attached to the rock by a calcium carbonate holdfast. Sea pens evolved from that group, and the base of their colony is an inflated anchor that can be inserted into the sediment. And then a subgroup of those sea pens re-evolve the ability to live back on rock, but they still have that fleshy base, and instead of cementing themselves, they suction cup themselves using that peduncle. It's fascinating. And the other moral of that story is that you can only adapt with what you have. You can only change the morphology you have. So it's not like the sea pen could say, oh, well, now I'm going to cement myself using a holdfast. No, I have a peduncle. How can I modify this peduncle to hold on to a hard substrate? Oh, well, what if I turn it into a suction cup? Oh, thank you. Flip a coin.
Depends on whether you think you're going to see something interesting in the next 2 minutes.
It'll show up right after I leave. Okay, I'll go to lunch first.
Okay, go for it. No, go for it.
Yeah.
Go.
All right. See you later.
So our fearless biology co-lead, Scott Francis, has gone to get lunch, and hopefully we won't see any biology for the next 15 minutes while he's eating, because then I won't have to try and wax and wane about what I see. Luckily, we do have a cohort of shore-based scientists that we're in contact with through a chat room and also on a phone line. So if anyone feels like calling in on a Sunday and giving their expertise, that is always welcome. And what we see now is more of just these coral skeletons that have littered the seafloor, and then also these occasional basaltic blocks, these pieces of rubble that have fallen from upslope. And while Scott's away, Ashton will play, and so what I'll probably try and do is make sure that we make some headway in terms of our transect to try and get a little bit more shallow slope. We started this dive at a maximum depth of about 1,075 meters, and we had hoped to get to about 980 meters, although that was a pretty long track length of about 750 meters. We've currently at about 1,045 meters depth, so yeah, not quite halfway up the zone we had wanted to go, but as you can see, there's an amazing amount of diversity and abundance in just a small footprint of area, so it makes getting further difficult. Joanna is saying in the chat that they have my back, so I'm relying on them for identification. So if they see something, say something and shout, or else I'm just going to fly right over it until I see the next beautiful rock. And so just to give a general context of where we are, we're on the Mid-Atlantic Ridge. We're about 340 kilometers north of the island of Corvo, the northwestern island in the Azores. We're just north of this leftward stepping transition, this transform fault in the Mid-Atlantic Ridge. So it runs north-south, and then it steps over about 15 kilometers westward and then runs north-south again. So we're right after that step, but not on the Mid-Atlantic. We're about 15 kilometers to the east of it. The Mid-Atlantic Ridge is a tectonic plate boundary. It's what we call a divergent plate boundary. So it's where tectonic plates spread apart. It's where new oceanic crust is formed. It's one of the three types of plate boundaries, the other two being convergent, where one plate subducts under another, and then these transform systems where the plates slide past each other. seafloor we're sitting on, the oceanic crust, not the actual visual seafloor we see in the camera, but the oceanic crust that we're sitting on, that was erupted from 14 kilometers to the west of us in the mid-ocean ridge. Right here, it's about 1.4 million years old. And you see just all of that marine detritus, all of those dead corals. Now, in this area, if I had to guesstimate, I'm guessing that the thickness of these sediments, which are derived from all this marine detritus, all these calcareous tests of things in the water column above us that fall down, and then they form this calcareous ooze, this degraded marine detritus that slowly gets cemented together. It could be upwards of 150, 200 meters thick in these areas before we get to that primary oceanic crust beneath it. These marine sediments are very weakly porous. Fluids do not flow easily through them. And so when we see these rock outcrops, they're important from the standpoint of they're the places where we expect lots of fluid exchange with the deeper crystalline basement. And so we're slowly just transiting up this slope, looking at the change both in the diversity and abundance of marine life that we see from where we started. You can see wherever we get one of these little basaltic pieces of rubble or outcrops, there's always marine life on it. They use it as kind of an anchor point. So they're little islands in the ocean. And if you heard that bell in the background, that's the ship testing its alarm system every day at noon. It's a nice little reminder to wake up. Now, a lot of these basaltic rocks, that surface that you're looking at is actually not the fresh basalt that was erupted at the mid-ocean ridge, at the mid-Atlantic ridge. That's actually coated in a manganese coating. And so manganese is a mineral that's naturally present in ocean water, and it precipitates out of the ocean. And it can precipitate out really in two ways, either what we call diagenically, so in the sediment, and it can form these little buried or slightly prone manganese nodules, or hydrogenically, in the water directly, and that forms on these rock surfaces. Now, these mid-ocean ridge systems, like the Mid-Atlantic Ridge, are really important because they were really kind of the coffin nail that helped prove this, the original hypothesis, the now theory of plate tectonics. So you can imagine when you have these really long zone of volcanic activity that's erupting these long linear features of lava flows, and those lava flows are being erupted, and then they're being pushed outward perpendicular to the ridge axis, so they're being spread laterally. And every time one of those eruptions occurs, the magma that is erupted is extremely hot, and it's above a temperature that we call the Curie temperature. And that Curie temperature, we have maybe a hermit crab in the video and lithoplacids. And right now, if you're looking on the camera, you're seeing an ROV team swap out. So they're doing an exchange of information for situational awareness of where we're going and what we're seeing. And then they'll swap out. These ROV pilots do 2 to 4 hour watches, depending on what's going on in the activity. And so we're just kind of holding still while they swap out. And so, as I was saying, these mid-ocean ridge systems are really important because they were kind of the coffin nail for proving this theory of plate tectonics. And so you can imagine all this lava being erupted on the seafloor and then spreading perpendicular to the ridge axis. And as it's erupted, it's really hot, and as it is seafloor and it cools below this temperature that we call the Curie temperature. Now, above the Curie temperature, magnetic particles, like things like magnetite, so these iron oxide minerals, in the lava are basically moving around in all sorts of directions. They're thermally excited, but as they cool through the Curie temperature, they take on the magnetization of the Earth's current magnetic field. And as they, so that permanent magnetization of the Earth's field, when it's erupted, gets locked in. We call this thermoremnant magnetization. And that magnetic signature gets locked in, and it stays there even though those erupted, now cooled lavas are spreading away from the mid-ocean ridge system. And then you can think of the next eruption that happens. That lava cools, and it gets a slightly different thermoremnant magnetization. It gets a slightly different magnetization locked in depending on the magnetic field. And our Earth's magnetic field is constantly changing, and it's occasionally reverses. And so when we have a reversal in that magnetic field, the magnetic signature in those lavas that gets locked in is opposite of what it was previously. It's in a reverse fashion. And so by looking at these variations, both in the polarity, the positive or negative, but also the magnitude of those magnetizations, we have proven that these systems are spreading apart, and they're recording a record of Earth's magnetization. And so we're still just traversing along. Our ROV team is still just finishing up their swamp. And they're kind of just going through the camera system. And we see another one of these sponges that we were just looking at earlier. To me, it's interesting the lack of marine organisms we're seeing now. But as I said, it's one of the primary controls on it is just the ability to have anchoring points, and so we don't see a lot of rubble. We don't see a lot of large basaltic blocks like we did further downslope, and so we kind of go through these small deserts of marine life, or at least kind of the macrofauna of marine life that we're used to seeing through kind of our normal camera lens. And that was the bird's nest sponge again that we saw. Thank you, Joanna. That might be another one, hard to see, just passing over the lasers now. Pheronoma carpenteria. No, that's okay. I was just... Yep. Thank you. And so while we're looking around, and we have kind of, we have a few things to guide where we're going. We have our general ROV track that we first designed about 2 days ago based off of the existing multi-beam bathymetry. We then have an updated ROV track that we do at about 5:30 in the morning ship time based off of our updated multi-beam mapping from the night before. And then once we're actually down on the ground, for lack of a better word, or on the seafloor, and we're undergoing our dive, we have our visual camera system that can help us see if there's anything of interest where we want to go. But we also have a sonar that's mounted on the ROV, and that gives us some idea of what might be beyond our visual extent. And that sonar can actually pan up and down, so it doesn't have to, if we're on a slope like this, we don't necessarily just see the sonar from right in front of us. We can pan it up that slope and see if there's any prone features or any outcrops above us. And so if you're listening to the ROV channel, that was the ROV navigator communicating with the bridge of the ship. And so they're telling the ship to move in a certain direction, at a certain velocity or a certain speed, and at a certain direction. And so that's going to move the ship, which is going to move Sirius, which is then going to allow the ROV to have a different little radius to operate in. And so Jana was saying in the chat there was a large sponge. I did not see it. We'll see if we can get eyes on it again. I think it's out of the frame above us. Luckily, if it's out of the frame above us, we're going up, so hopefully we'll come across it again. Yep, thank you. Oh, I think that's the large sponge they were probably talking about. And so I think this is the same one we saw earlier. Yeah, that had that outer covering with the shrimp listening and looking in it. Yeah, this one seems to have that same covering. Like from my understanding, listening to Scott and the shore scientist, that covering may act to trap or protect, depending on how you look at that coin, a shrimp species that live inside of it. It is the sponge is a euplectid. EUPLECTELLID. And another urchin at the base of it. Actually, can you look at the top of the sponge again? like we did on the, yeah, just, yep. Want to see if there's anything else inside this one. Oh, Tara Luke is spelling it phonetically for me. Euplectelid. Thank you, Tara. Yeah, trying to see if we see any creepy crawlers inside of it, but it looks like a long way down, maybe a hard angle, so, yeah. Yeah. No, that's great. Thank you. Woo. Extreme close-up. Yeah, and let's take one more quick look at the base. Yep, thank you.
I heard tell you were looking for some creepy crawlies. Nice Euplectella or Rigodrella. Looks like a Okeanos bamboo coral there at the base, and an urchin, shrimp. And I was noting while I was eating my lunch, I was watching on one of the many monitors placed around the ship that we were going over a lot of coral rubble. The environment seems to have changed a little bit. I think what we're looking for is how the sponge attaches to the substrate, if I understand what I'm seeing in the chat room here. It's at a tough angle. I'm wondering, when you're ready, if you have more zoom to go in, I'm wondering if that's an organism or an old holdfast, that round disc-like thing, white disc-like thing at the base of the sponge. Yep, correct. Round discate base. Okay. Wow, yeah, that's a limpet, or somebody's going to tell me that's a monopaclophorid and we're going to go crazy. It's definitely a mollusk, and it looks like I see little tentaculate-like structures coming out around there. Almost looks a bit like a keyhole limpet. There we go. Good view. Nice. Oh, yeah, that's great focus. Yeah, that's a keyhole limpet. I can see the opening at the top. There would be a current of fluid that comes in at the head end. Water would pass over the gills and then go out that little opening that you see at the top center. Excellent. Thank you. Maybe a brachiopod to the left of this beautiful Okeanos bamboo coral. I was noticing while having lunch that we were passing over this rubbly area, and just like right here, two more Okeanos. We just passed one by the sponge. So the Okeanos are doing just fine in this sort of loose aggregation pile of coral rubble. That's not a very stable habitat, so many species wouldn't be able to grow here. So, you know, further down the slope, we saw those really large black corals, bamboo corals, some of the Lophelia. They wouldn't be able to get good traction on this sort of unconsolidated bottom. So it's kind of interesting. And I'm not sure if this is stuff that has grown in place and died, or if it's tumbled from further upslope. On our first dive, we were seeing lots of rubble of Eguchisamia, and then when we got towards a little higher on the slope, we started to see it live. I think that's a little Chrysogorgia that we're passing over. And they do tend to require a little bit more stable substrate. Another one of these euplectellid sponges on the upper right, and an Acanella. We're just passing through 1,025 meters depth. When we landed, it was 1,072 meters, so we've only gone up the slope 50 meters. We've gone, I think, horizontally some distance. It wasn't steeply sloped where we first landed. And that was at about 09:45 ship time. So about 3 hours. 120 meters horizontally. Thank you, Nav. Let's have a quick look at the sponge upper right. Just going off the screen. Yeah, this one looks different to me from what we've been seeing today. It's interesting. It looks like it has a couple of those sort of chimney openings. I continually have a discussion with my sponge colleagues about whether these are still called oscula or have some other name. You can see the lasers there about 10 centimeters apart, so the diameter of that sponge is about 10 centimeters on the upper part anyway. So I think this is another glass sponge, and it has this fibrous attachment to the seafloor. Getting a good look at it right there. Joanna says she's checking her notes and her monographs to see if we can come up with some ID for this. And again, just a reminder, so this glass sponge, Hexactinellida, is the larger group to which the glass sponges belong, produce silica spicules. And so all these little filaments that you're seeing in the skeleton itself are made of silica. And the cells, which of course we can't really see, the tissue, are draped on that. Lots of very small brittle stars also clambering up this sponge. Maybe some kind of little worm just below that lower ossicle, the opening. So that would be for the X-current flow. The water that has already passed through the sponge walls would come out those openings. And we have a little chirostylid crab, I believe. Maybe that's not... I'm not sure if that's a chirostylid or if it's something else. It's a squat lobster of some kind. Let's see if some of my colleagues can identify that crab. I'm ready to move on. Thank you. Carlos, who's back in Horta in the Exploration Command Center on the Azores, notes that in the dives done in this area, they saw fields of Acanella, this yellow-orange bamboo coral. There's one, the pinkish one in the far left, along with Chrysogorgia. So mixed fields of Acanella and Chrysogorgia together, especially in the flatter areas, and both at similar densities, down to about 1,200 meters. So we're seeing a lot more of the Acanella today. We've seen a couple of the Chrysogorgia, but not as much as the Acanella. If that eel-like fish stops, you can zoom on it, but if it doesn't, just let it go. They're fast, aren't they? Yeah. So this one, if we can zoom on this bottle brush just left of center that's coming into frame at the top, it's kind of grayish color, almost in the center now, just above the lasers. This might be the Chrysogorgia that Carlos is referring to, that we have both Chrysogorgia and Acanella together. Yeah, indeed. So this is this bottle brush type of Chrysogorgia. It looks like it also has a squat lobster in its branches. One of the things that's interesting to me is the Acanella and this Chrysogorgia have a grossly similar overall colony morphology. I don't know if it's something about living in these sort of rubbly habitats at this depth. Yeah, there we go. You can see all the branches comes up in this really complex pattern. The branches aren't too long, but they create this kind of neat internal space like a cage, and we very often see either a shrimp associated with it, or in this case a chirostylid crab or squat lobster would be a common name. They seem to take refuge on the inside of these branches. We often, I don't see it on this one. Oh, there's a couple of amphipods. I can see their red heads just down below, just went off screen. We also see associated with these Chrysogorgia colonies cerate octopus eggs, the common Dumbo octopus. And they also, the analysis suggests that they are deposited in towards the central axis, which suggests also that they're being kind of protected by the coral itself. Nice video. You can see lots of these amphipods, their eyes are reflecting the light, so they appear bright white on these red heads. Is that a big sponge? Science is happy when video's happy. So just a note about that sponge that we passed there. There's a suggestion it might be in the genus Ascanema, although Manuela says she's never seen one with two oscula. And Joanna said something similar was observed in Orphan Knoll, which is over on the coastline by Newfoundland, not connected to the Mid-Atlantic Ridge. Yeah, there might be a large sponge on that rock Caitlin was pointing at on the right. Oh, okay, okay. Yep, yep. Copy that. There's a big rock just coming up ahead, and there's plenty of things growing on this rock. Oh, let's get a zoom on the fish, please. There's two fish there, actually. The left one, yeah. I said left, but I think the right one's a hake. So the left one is a rat-tailed grenadier. Oh, okay, we'll go the right one. No, that's no worries, no worries. Yep, it's good. I think this is a codling. I think we got some images earlier, but this is a much better image. Got that really tall dorsal fin. No, let's go to the next one, please. There we go. So this is definitely a grenadier, so-called rat tail. Lots of species of rat tails. Okay, that's good. This one has a very rounded snout and head. It's passing over a cup coral there, that brownish-orange structure. Come on, turn towards us just a little so we can see the front of your face. You can see the white line along the body is the lateral line. There would be sensory cells in there so you could detect motion. Very rounded head. Oh, that's great. That is great. And the fish is being very cooperative. You can see it's got that ventral mouth. You notice the mouth is sort of tucked under the head, and so it probably comes close to the bottom, opens, and sucks up its prey. Good stuff. Great work, team. That was very nicely done. Looks like a couple of rocks here, and you can see again when you get to these local highs how much the fauna changes. You know, now we have this stable, this looks to me like another one of these pillow-like cone features that Ashton was describing earlier, and this bottom is much more stable than where we saw all that coral rubble debris. And so as we move up on it, you'll see there's a lot of stuff growing on it. Can we have a look at the yellowish stick that's on the left of the lasers? I saw this earlier, and I'm really not sure what. The first time I saw it, I thought it was a crinoid stalk without a crinoid at the top, but I no longer believe that. Yep, your lasers are just about to hit it now. I wonder if it's a black coral. Yes, indeed. I believe this is a black coral. Yep. So it's a black coral. It's hard to tell that there's a whole string of polyps moving along that skeleton. They all seem to merge together. It just looks like one long thing with a lot of tentacles. But those tentacles would be arranged in sixes, and within each ring of 6 tentacles you would find a mouth. So this one's got a couple of crinoids sitting on it. There we go. There's a good view. I guess, I mean, I don't actually know. I guess this would be in the genus Parentipathy, or excuse me, Stylasterin. But I'm not 100% certain of that. And a really nice view of this crinoid, and I can see it looks like maybe there is a snail on the back of one of the arms. They do have these little predators. Yep, there's a second one up on the top side. So these are specialized snail predators of crinoids. Or there's another crinoid. So the crinoids are just perching on this relief basically up above the bottom in order to capture stuff out of the water column. They could release themselves from that coral and swim elsewhere if they so chose. So down here at the bottom we're seeing at least 3 stalked barnacles and several hydroids also growing off of this skeleton. And then on the rock on the upper left there's a calcareous tube with the tentacles sticking out. I was going to say that, I mean, that should be a type of fan worm, but it almost looks like an anemone sticking out. It looks like a serianthid anemone, which is a tube anemone, inside a serpulid polychaete worm tube. but maybe it's a serianthid tube. Huh. How about that? That must be what that is. So I must have misinterpreted. That might be a kind of soft, leathery tube that anemone has secreted, and you can just see its tentacles sticking at the end. I think that's what that is. Well, hopefully I'll get some input from shore and find out if that is a worm or an anemone. I want to say it's a worm, but my eyes tell me it's an anemone. Welcome back.
And there's no way it could be a worm anemone.
Not that I've heard of,
Okay.
but we could always describe something new.
Yeah.
Maybe that's a good segue into my finishing the question that you asked earlier about those Latin names. Why do we use such complicated names? And so, we settled on Latin. And I just want to point out, you all remember from your high school days, we have this hierarchy of naming: kingdom, phylum, class, order, family, genus, and species. And there's a very specific set of nomenclature or rules for using all of those names. But every species name is basically a hypothesis. And so it's a hypothesis that a scientist said, I believe that this name represents this suite of characters on this organism. And that has to go for peer review and is constantly being reassessed with new data. So we're on another one of these nice pillow cones, Ashton. You can see that once we get here, the characteristics of the community completely change from all that rubble. Well, look at all the stuff taking advantage of this. It's great.
Now, I asked the other day if you had a mnemonic, and Google tells me that one of the mnemonics is, Dear King Philip came over for good soup.
What's the D?
Domain.
Domain, right.
What do you mean, what's the domain? You're the taxonomist.
Yeah, I know. My bad.
Yeah.
We didn't really, when I was in high school, we didn't talk about domains. Kingdom was enough, but then we started to discover all these interesting bacteria, and we started using domains. Wow, look at this. How high are we on the slope? We're not at the top yet. We're getting close, though, folks.
Yeah,
we're getting close
years.
to the top of this little ridge, and look at how much the density of sessile organisms has picked up. That's a very interesting sheet.
That almost looks like a thin layer of a sheet flow, but actually I think it's probably just more of a... I don't think it's a sheet flow just because of how thin it is. You can imagine something that thin isn't going to actually flow underwater like that. They should be much thicker. So that just might be a kind of a crusted, an incrustation crust. So, part of the seafloor that's been lithified or become solidified as a crust and then later broken out.
So this is looking great. We've been talking earlier in the cruise, I think actually before we started diving maybe, about what constitutes a high-density coral sponge community. We may be getting into one, and it has to do with the number of organisms that you see per meter squared. And it would be even better if the density was high, and I'm seeing density. I meant diversity is high. So high density plus high diversity, that means you have a biodiverse area. There's clearly a lot of food, and so there are a lot of organisms also that are surviving. So,
what are these little fish on the bottom right, and then I think also in the middle?
Those are Oreos.
Those are the Oreos.
Yeah, the Neocytus.
I just had some Oreos with lunch.
I passed them up.
Oh, yeah.
I confess. I eyed them. No, you can ignore that. That was just Ashton asking me. Yep. Yeah, I'd like to have a nice view of all this diversity that we're seeing on the left side here. So we pass over it. And maybe as we pass over, just get a little tighter once in a while. So thank you. So these interesting sort of Dr. Seussian branching structures, those are bamboo corals. I'm really intrigued by this one that's in the foreground. We had this discussion earlier about, what do they look like when they're younger before they get big into that sort of ecnomysis size, but I suspect that's what we're looking at. There's lots of Leopathes black coral are showing up again, especially in the background. The beautiful yellow sponge. In fact, I don't think we've looked at that yellow vase-like sponge yet. Yeah, that one in the upper right. We saw it like the Hertwiggia before.
Yeah.
That had the same sort of color and texture, but it didn't have this morphology. So one thing I don't know, maybe the sponge biologist on shore can help explain to me. does that morphology grow up into this beautiful vase? Were we looking at an early stage, or is this something completely different? It's gorgeous. Oh, apparently, so this is Hertwiggia falcifera again. So that is what we were looking at before, so very cool. And stand by, Carlos, we will be zooming. Fan-shaped organisms at the back.
So with regards to this Hertwiggia, the one we saw earlier was earlier. It was younger, but I guess I don't have a sense of, what's the age difference?
Yeah, it's a good, excellent question. Yeah. I also have no idea.
Yeah.
So Joanna says they've never been dated, so not aged, so we don't know. We are, the number of things that we've identified that would be useful to have an in-situ camera just sit there for a few years to help us answer some of these questions.
A few decades.
Yeah, exactly. Pilot, I'm ready for another request if we're done with video. Okay, thank you. To the left, just behind, just swing left and I'll point it out to you. There's going to be kind of a fan-shaped colony there. Just coming into view, it's got that large crab on it. If we could get a look at that. Thank you. So the crab apparently is Sternostylus formosus, Carlos tells me. And I have to do...
Sternostylus, does that mean it's got a large back claw?
Oh, that's a really interesting question. So that would make sense with respect to this group of the chirostylids, these squat lobsters, are adapted with those back legs to hang on to corals, and they're often very hook-like. So absolutely, that could be. Good call. The chirostylid, I know that family level name, which is a higher level name than the genus, is describes the long pointy arms with the pincers on the arms. So this is another black coral colony, and Carlos Dominguez Carrillo down at the Exploration Center had asked us to get a good view of this. Oh, I don't recall. Is this a Parentipathy, or is this another just really densely branched Stauropathes? I love the crab waving his long chelae is the technical term that you would use for those claws, and so the legs on which the claws are called chelipeds.
Wave your claws in the air like you just don't care.
There you go, everybody. So we're hearing that this is the genus Dendrobathypathes. Dendrobathypathes. So it's the first time I think I've said that. So that's great. A new addition to the diversity. And these little thumb-like structures that are coating the rock just underneath it, those are zoanthids, the colonial anemone, and they've all sort of closed up and retracted their tentacles. Carlos is now trying to disown all ownership of that crab name.
I saw him backtracking, but I wasn't going to say anything.
Yep. Carlos, we appreciate taking the gamble early, throwing out a name, and then we check it out. There's a just a quick zoom on the right. You see that pink? You don't have to move the RV. The pinkish colony on the rock behind. Thank you. I'm wondering if that's a Paragorgia back there. I haven't seen any Paragorgia today. It is. How about that? No, it's in the rock behind. There we go. That one, the branching one. Yep. Bingo. So that's a bubblegum coral, and you can see why it's called bubblegum coral. It's got that sort of texture and color. Most of the polyps are retracted right now. The very top right, I see a couple of polyps with their tentacles extended.
I feel like we've run across a lot of things with the polyps retracted.
Yeah.
More so than in the other dive, maybe.
Today, you mean?
Yeah.
I see.
Anything that's environmental or just?
That's a really good question. Yeah, it's a feeding, right? They're going to be out for feeding. I don't know.
Yeah.
Is there a resting time period? If they do that, is that months?
So their resting state then is them retracted, or I mean, what's a?
I don't know.
Yeah.
I don't know.
A lot of I don't knows here.
I would say that it's great that there's a lot of I don't knows.
It is
means
exciting.
that we're throwing out challenges there for students to say, I'm going to answer that question. Anyway,
yeah, I mean, if
there's no energy, right, to having those tentacles extended, so that could be a resting phase. But there's a lot of incentive if some fish is coming to nibble at you to pull it in.
Yeah, if we were looking at things where we knew all the answers, I guess it really wouldn't be exploration. So.
Absolutely, right, exactly. Yeah, so I think we're at the top, or pretty close to the top of this ridge. Okay. So Pilot says that this may be a local high, and we'll sort of, pursue it and see if there's anything beyond that's a little bit higher. But, a little bit more distance. I'm looking at this 3D perspective map. It may be time that we'll turn towards the north after we've scaled up this slope. But what I'm seeing is really promising to suggest that for the rest of the dive, we may be seeing this kind of density of suspension feeding organisms. Fantastic. Maybe we could get a close-up of the white barrel-like sponge that's on the lower left. Thank you. I'm not sure I recognize that from earlier today. Yeah, 7 o'clock. Thank you, Brian. I should use the clock terms. So we're zooming in on the sponge, and all the stuff that you see around it is Sclerexsonian coral, so true stony coral. Mostly, I think, Lophelia, the genus Lophelia. And I have no idea what kind of sponge this is I'm looking at. Joanna Jevia says that this is a demo sponge. We've been looking at a lot of glass sponges today, so this would be the other major group of sponges. Those pinkish thumb-like structures are zoanthids again. Some of these Lophelia colonies up here are a lot bigger than some of the others we've been seeing. That sponge looks like it's almost half a meter across. So just kind of listening to pilot and navigator talk, the plan here is we'll just sort of skip over this feature that we've just come up onto from climbing this slope, and we'll see if it continues or if this is the sort of local high, and if it is, then we will move along the length of it, basically going to the north. You'd have to consult your overnight dive plan or look at the quad screen, which shows the navigation map in the upper right corner, and you can see that we've been crossing these contours during the day. The sort of yellow-green part of the map up towards this top, much flatter peak, and once we've acquired this top flat peak, we'll move along. We'll sort of turn to the left on your map, the upper part of the map, which is facing north. I'm intrigued by that tall yellow colony. If we could, yeah. I do understand we're about to leave. Okay. Oh, I gotcha. Pilot change, no problem. That's the one. Thank you. And for onshore, not for pilots, I'm not positive. We'll see, because we're going to cross over this thing. But in the far right, I'm not sure if that's a big planar purple colony or if it's just it's white and in the distance it looks purple. So we'll see as we get there. A lot of cup corals up on these rocks here. All right, so we're going to get a zoom on this large yellow colony. I was wondering if from a distance this was another one of the Acanthogorgia. Beautiful framing of this coral by the ROV pilot. He set the ROV down so we can look up against the black background, and it really makes the colors and the shape pop. It's beautiful. Yeah, I think this is a plexaurid of some kind. It's not an Acanthogorgiid, so I would venture to say that this is the first time today that we have seen this particular species. Very interesting. It's got a lot of brittle stars on it, several crinoids. Yeah, thank you, pilot. I was just corresponding with Shore. There's various interest in sampling various things, and I'm just pointing out we have one box left, and so we need to have some good consensus. Yeah, a view of the base would be great. So this looks like it began its growth on a Lophelia, the Stony Coral, and then took off from there. Very interesting. You know, before you tilt up, I apologize. A really neat view right now of those polyps that are extending off into the black on the right side. They look like they're coming out of a little horn. So those must be hydroids, and that's a good view of what I was describing yesterday as the perisarc, which is like a chitinous covering, a tube that the colony secretes, and then the soft tissue is on the inside. And these are what we would call athecate hydroids because there's no cup that surrounds that feeding part of the body with its tentacles. That's a great view. We don't often get it from that angle. Thank you. You can move on now with your scanning upwards. Also, two gastropod snails here on the base of this yellow colony on the left side. So what we're going to be doing now is getting some nice video, and we're going to be panning upwards. Yeah. So, Chris, pardon me for a second. Sorry about that. So there's a lot of interest in this yellow coral. I think I'm seeing another one on the far side. What do you think is the chance of being able to clip a small branch and then slurp that small branch, as opposed to going into the bio box? I'm pretty certain the one on the far right is the same thing, so whatever one is positioned better for you to work with is fine. Yep. If I'm saying left, I mean right. I don't know what I'm saying anymore. Yeah, that's fine, Chris. The branch points are not important for this one. Correct. Yes, and Joanna Javier is basically saying the same thing. She writes a stunning deep seascape, and it really is. And one of the things that I really like about this is we're seeing diversity, density, and this array of color. And I think a lot of people don't appreciate the color that deep sea organisms have. You think, you know, it's dark, there's no light, there's no color. That's wrong. There's that purple octocoral again. How interesting. If you can, yeah, absolutely. Okay, understood. Copy that. Well, this was a good opportunity to see that stunning seascape and now to see this purple one, so it worked out just fine. So this is an interesting one. You notice that the branches have a much smaller diameter. The polyps are, I think, all retracted. I would guess that this is yet another plexaurid, which that name is a family level name, so the Plexauridae. There's a lot of genera and species in there. They're really hard to distinguish from these videos. Yeah, you can just see the polyps there. The tentacles are mostly pulled in. That's probably what this is. Les Watling will know, when we were on the New England Seamounts, we found this magnificent, huge purple sea fan that was kind of similar to this. And I think still to this day, we did that cruise in 2005, we're not sure what it is. So they're tough to identify even when you have them in hand. There's another interesting thing about them, and that is that they have an interesting biochemistry associated with them, and many of them, for whatever reason, are really hard to work with their DNA. And we think it's some other chemical compound that they have is inhibiting our ability to either extract the DNA, or when we extract it, it co-precipitates with this other compound that then interferes with downstream reactions. So, for example, it's a lot easier to work with bamboo corals and the chrysogorgiids and the sea pens in terms of their genetics. Just makes the whole job a little more challenging. No, we're fine with that, Chris. Thank you. Yeah, this is really good stuff, and this is one of the reasons that we like to try to get to the tops of these ridges, because this is the sort of scene that we're hoping to see. It was interesting on dive one, we did get to the top, and yet we didn't see any. All we saw was a big pile of rubble that was mostly dead. It's quite fascinating, and so yet another one of those questions we can put on our long list is, what's the difference? Why did we see it there and not here? Was that a salp chain maybe, or
Maybe, yeah.
siphonophore in the background? Chris Ma is wondering if that sideroid urchin that you see on the lower left is feeding on a coral over there. I've seen several sideroids in this patch, Chris. There's certainly plenty for it to feed on. Nope. Anything, whatever is easiest for you, won't matter for us what part of the colony is clipped. I kind of wonder if that branch hanging off the right is in an easier position to get something that's elongate to suck up later. Little twist should do it. There we go. Really nice, delicate touch there to try to leave most of that colony undamaged. Just take a little piece that we can use for genetics and identification. Now, the challenge will be getting it to the suction sampler. And again, on shore, you can look at camera 3 to see multiple angles for this collection. It's there. Yellow plexorid. Nicely done. There it is. We got in the canister. That was brilliant.
You impressed all of our shore-based scientists,
Yeah,
Pilot.
you got a lot of fans. A lot of fans there. Huh. Is it an amphipod or something? Looks like we also got an associate with that collection, an amphipod. Chris, before you leave here, can we get a view of the urchin? That's just, it's almost in the center right now. I'm not sure if that's the same one, but oh no, the one he was asking for is actually the one on the far right, because he wants to know if it's feeding on the coral. Thank you. Yeah, you go ahead and zoom. I'm not sure if that's an anemone or a cup coral. I think it's a cup coral. I'm seeing those white lines around the margin. Those are probably the underlying septa of the skeleton. There's a caprellid shrimp just below. I love those caprellids, skeleton shrimp. Yeah, I think that's a cup coral. So good eye. Is it in one or is it in two? The amphipod. Is it with the coral or is it in a different one? So, Chris, as we zoom in here, it looks to me like this sideroid is on dead Lophelia, so I would suggest that it is not feeding on live coral tissue, but is instead grazing perhaps bacteria or some other fouling organism that is on that coral. That's what I'm seeing there. Thank you, pilot. Cindy Van Dover makes a really good comment here, and I didn't say it well enough, so I'm going to read what she says. I think it sets a good example to sample so precisely without trashing the organism and environment, and this is something I really admire in the ocean exploration program. A big contrast from the early days of alvin exploration. So well done. Kudos, pilots. So before we did that collection, what we were doing, what the pilots are going to do is just kind of move over this feature that we came up onto and see, looking in the blue view. I can never remember the name of that thing. I always have to look over. Looking into blue view sonar mounted on the front of the ROV. Do we see any higher points in the distance, or are we basically at? Is this a local high, or is this the top of the ridge? And then we can make a decision on where we want to move further east or turn to the north. We're certainly on a beautiful feature here. Yeah. Okay, so I think we're happy just to now. Let's just follow the features in front of us if we're, sort of more or less on the top. Thanks. Pilot Watch Lead 2. So I'm seeing a really large, what looks gray from this distance, fan in the background there. We'll have a look at that. But then also I just want to let you know, in the front of it is that sort of brownish, reddish colony. So that's the Stylasterin black coral. And I've had a request, if we see large ones of those, to get lasers on the base. And that way we can start to get a sense of the ages of some of these different black corals up here. Thank you. It doesn't. I don't think we can do this one. It looks like it's kind of covered, but I just mean we're going to see several as we go. Yep. Yeah. So these brownish red ones, those are the ones we want to measure the base. But that large one, I just want to have a look at. I'm not sure what it is, other than it's large. It might be this large purple plexaurid that Les and I were, or have being discussed in the chat room here. That may be similar to what we saw in the cornerized seamounts, which aren't too far to our southwest. And can we have the lasers back on, please? Thank you. This Oreo is patrolling its garden of coral and sponge. And I apologize, Manuel, I missed the Paragorgia. I did see a Paragorgia earlier. And Stylasterin, I'm not sure what Stylasterin looks like. I don't remember that one. So I'm going to have to look it up so I know what search image to have. Yeah, it depends on the size of the polyps. I imagine it's not great for... oh, I see. I think so. This may be what Manuela Ramos was referring to as Stylasterin, and if that's the case, then this is going to be a black coral. It'll have very tiny polyps. Yeah, that fan. Yep. It's not one that I'm familiar with. Well, it's definitely not an octocoral, I can tell you that. So, yeah, I believe this is probably the Stylasterin. Yep, that is correct. So, yeah, interesting. This is another black coral. And so I said earlier today that the tissue, the soft parts of black coral take on a whole variety of colors, and it's the skeleton that gives it the name black coral. So here we see one that's kind of got a bluish, purplish hue to it. And the polyps are so small that you really can't even make them out. So you can't really distinguish the individual tentacles. You know, I can on some of these exquisite cameras just get a sense of some of them. But otherwise, no. This is a large one. I have no idea about the growth rate. I don't know that anyone has ever tried to age that. And that was a beautiful change in focus. And now we're looking at another kind of black coral, the Stylasterin that we've seen several times today. And we've noted that as far as we know, this is the oldest animal on Earth, or an individual of this species, or this genus, I should say. It was, I think, Stylasterin glaberrima. This would be a different species. It's got several basket stars with their arms kind of wound around the branches, type of brittle star, ophiuroid. I'm not sure if that's that. Oh, that's another brittle star that we see the arms of there. You can really see the black color of the skeleton. Yeah, it's tucked underneath there. Yep. Well, that was beautiful. That was great. See a little one of those goniasterid cookie cutter stars off to the lower right. Lots of diversity here. Another one of the bottle-brush Chrysogorgia on the left side. Plenty of Lophelia. This is a true deep-sea coral garden up here. Lots of diversity. There must be lots of food in the water to support all of these colonies that have probably been here based on their size, for many, many decades. And they grow so slowly that, damaging this with any sort of equipment, it would be hundreds of years, maybe thousands of years before it could all be replaced. We don't need a closer look. We've got a good identification of that one. We've got various requests for getting lasers on the holdfast, kind of what I've told you. I'm explaining that we don't have the right angle to do that here. So I think we're good. Thank you. So this heading that you're seeing on the camera right now, the ROV is pointed just east of due north. It's 45 degrees. Now it's pointing back. We're at about 60 degrees now. So pointing back towards the east, which is the dominant direction that we've been moving up today. Looks like a very large plate sponge up ahead. Looks like another of the cusk eels there. Yep. Understood. Okay. So I'm sure you'll have periods of where it's pretty dark on the screen, and that's because we have to go downhill a little bit. It's difficult for basically the ROV. We don't want to back down the slope, and so just stay patient while we go out a little bit into the water, and then we'll come back down and get the good images again. I'm really intrigued by this whip-like coral that's standing off the rock to the left, and then you see it's got the branches at the end. So one of my master's students, Mary Durie, is doing a project. I would love to. Yeah. Mary Durie is doing a project on this sort of structure of bamboo coral in the Pacific using all of the Okeanos Explorer imagery from out there. And we've been lucky enough to get some samples. And what we're trying to understand, I've said this a few times now, is how do these colonies change as they grow? And how do you know that, this smaller, only a couple of branch thing is the same as this larger multi-branch thing? Like if you look at the top of the rock, there's something there with multiple branches. Is that the same colony as this one? We know they're both bamboo corals, but we don't know they're the same species. So ultimately what we have to do is keep getting good images. We have some samples, we do some genetics, and then correlate that with what we can see in the images on these tight zooms. And this is an interesting one because you could imagine it would grow as a whip for the first part of its life, and then when it got to, let's say, a meter long, only then does it start putting out all these branches. That's a really large Okeanos at the top of that rock. That was kind of cool. There's a pretty high density of polyps on this skeleton. To be perfectly honest, I'm not seeing any nodes yet, but I don't know. Maybe this is not, no, it's not a bamboo coral, it's purple. Huh. Well, that's why you do the close-up. This looks like that purple one with the yellow polyps. I've just never seen a plexaurid that's had a long, unbranched stem or stalk that then starts branching later. And it must be something to do with the local hydrodynamics of where it's growing, but that's fascinating. Well, that's an entirely different morphology, colony-level morphology for a plexaurid than I've seen before. So maybe I should ask Carlos and Tamo Marina if they have seen this sort of morphology before up here. It's so unusual. Yeah, good. I'm glad you got us to see the holdfast there, that yellow sort of disc-like structure at the base that is attached to the rock. This is totally unusual. And I do think it's the same as the one that's at the upper right. But maybe this is telling us something about, you know, once the larva starts growing, then the local physical conditions are going to dictate, you know, when do I start branching, as opposed to being genetically programmed. East to west. Let's see. East, so that would be coming over the ridge that we've been climbing from the other side. So it's coming into your face. Oh, cool. Okay. So it sounds like the current, at least right now, is coming towards us, and these colonies are all well positioned to capture it on this ridge that they're living on. Chris, now that you've made all your plans, you'll be not surprised at all that that thing is so unusual people want to collect it. But if you tell me you've already made the move to move, then yeah, it's extremely unusual. It's not a growth morphology we've seen before. Don't know what it is. Excellent. Glad I got that in quick enough. So, Pilot, this is going to be much like that last one that you just sampled. It's kind of arbitrary where you clip it. There's no way this is a bamboo coral, so the nodes aren't important. But beyond that, we really don't know what it is. Purple plexard. Yeah, I mean, if you feel pretty good about that, it seemed to go pretty well last time, I thought, and it just preserves that bio box since this is kind of flexible and we can get a small piece. Copy that, thank you. Arvin, this is 7B. Yeah. No, this is not stiff. It's quite flexible. You should be able to cut, and if it doesn't cut right away, a little twist would help. And what you pointed to was a good piece.
You're muted.
Thank you, everybody. He clearly didn't need my help. There goes one, there goes two. You guys are good. There it is. It's in the canister, number 2. There should be 2 pieces down there. Okay. I don't know why there's some kind of error on that collection database. Yeah, oh yeah, I've got it coming back in. Thank you, Fernando. I've done that a few times. Get used to it. It's just frustrating because it's off on the timestamp a little bit, and I have standards to maintain back here. So that was outstanding, pilot. Before we leave, if we could just go back down approximately where we were, I'd like to do 2 things. One is to fire a Niskin while we're in this really high diversity area. and the other is to, there was something in front of what we were collecting, and if we still see it, then we'll get a zoom. If we don't, that's fine. We'll look for others. Yeah, it was right at the base of that thing you just collected. Yep, so if you just go in forward there. Thank you. It's like a fuzzy whitish bush that's on the lower right of what you just clipped. Yeah, there's two of them. The one that's dead center is just fine. Yeah, I think they're both the same thing. Okay, I think we had a good call from shore earlier because I just assumed that this was a hydrozoan from a distance, but a couple of my colleagues, Les Watling, Asako Matsumoto, I think, suggested this might be Thuarella grasshoppii. I think Les said that first. That's a primnoid octocoral, belongs to the family Primnoidae, which I'm frankly surprised is very, very common in the deep sea, and we've seen very few of them so far in this expedition. And I'm pretty certain that that is what we are looking at here. Originally described, I think, this species from the New England Seamounts by Stephen Cairns, very prolific octocoral and Sclerexsonian taxonomist. Yeah, good stuff. Yes, we do. Please feel free to do it right on the bottom. So. Yeah, if you just stay focused on the, I want to count 1, 2, 3, 4, 5, 6, 8. Yeah, okay, thank you. So I'm good here, video, whatever you want to do. Okay, thank you. Oh, is it Niskin 3, I think? Thank you. Yeah, and so with all the diversity down here, we just took another water sample for eDNA. So this looks like there could be potentially plenty of sequences that are possible to detect. The large plate sponge just on the other side of that rock. Yep, thank you. And pilot, can we have the lasers back on as we look at the sponge, please? Thank you. Huh, look at that. This sponge is growing over a coral. And yeah, I'm not sure who got there first. I think it's a Lophelia type coral, but look at that. I think they're hovering above this large sponge is another purple plexor that looks like it has a long stem without branches. So that's kind of interesting. Look at that. It's like this Lophelia is wearing a sponge as a cape, or it's in its own little amphitheater or beach umbrella, I suppose. The beach umbrella sponge. Well, that's definitely what the coral is. So that's a Lophelia, Sclerexsonian colonial stony coral. Seems like it's doing just fine. So we may be going for a beauty shot, but we are more interested in the sponge than the coral. Joanna offers an identification of Vulcanella, which would be a type of demo sponge. Again, demo sponge is this very large group within the Porifera, the phylum of sponges, where most sponges would be classified. And so it's a very general sort of grouping, but it distinguishes it from those glass sponges. The demo sponges still do have silica fibers in them. Just don't have the same kind of construction. All kinds of barnacles as well on that coral tucked under there. Asako suggests that's Madrepora and not Lophelia, so Madrepora would be a different genus of these branching stony coral. Yeah, no, I'm good with it. We got an identification, so I'm ready to move on.
Yeah, so we're at the more or less local summit of this normal fault that we started our dive on. We started our dive on at about 1,075 meters below sea level on the southwestern face of this normal fault, and then came up about 100 meters to the top of it. That's a little unclear from our bathymetry how much more we have to ascend. But you can see this long exposed ridge of pillow basalt fragments. You see this shrimp dancing through the water column. And this exposed ridge of pillow basalt fragments, given all the currents that are coming, the ocean bottom currents that are coming up and over this ridge, it's pretty much swept clean most of the marine snow and detritus from this area, and this has given a lot of exposed rock for these corals and sponges to adhere to. The strong currents also provide a lot of opportunities for them to feed. And so we're going to continue along this little exposed ridge, looking at these communities and maybe seeing how far it extends. Can we get the lasers back on when you have a chance, please, too? Thank you. Yeah, it's really amazing how quickly everything below maybe the top 2 or 3 meters that's out of this more exposed surface current, or ocean bottom current, how much more sediment and marine detritus there is. And it looks like we might have another large outcrop ahead of us, about 30 meters out on the sonar on the blue view. I'm guessing it's kind of a continuation of this ridge and then another of those large pillow fragment outcrops. And so if you're just joining us, we're about 340 kilometers north of the Azores. We're on a normal fault that's just north of a small step over in the Mid-Atlantic Ridge. Mid-Atlantic Ridge is extending northwards, running more or less north or south, and then gets to about where we are, and it steps over as a small transform fault to the west. It moves about 13 kilometers westward before it moves north-south again. And so we're just a few kilometers north of the beginning of that step over, north of the eastern portion on oceanic crust that was erupted about 1.4 million years ago. And most of the area that we've been seeing has been characterized by lots of marine detritus and marine snow and coral skeletons. I got a geology lie for you as an answer then. So the way we can really tell is from the bathymetry that we collect beforehand, and then our understanding of the general structure and morphology of these mid-ocean ridge systems. So the Mid-Atlantic Ridge, more or less, is oriented north-south with that long trench that we see in the general geographic background of the bathymetry, with this valley in it that's about 8 kilometers wide. And so the feature we dove on the other day was this substructure within that valley, so right in the center of it. And those volcanoes tend to be very elongate in the north-south direction, ridge parallel, and then really compact in the east-west direction, ridge perpendicular. So it's a topographic eye. It's right in the middle of the ridge. It's a pillow volcano. That's kind of the features that we look for. This is, outside of that rift valley, but in this tectonically active zone. And this feature is a little strange because it's a little older and it's a little bit more eroded. But in general, on the side closest to the ridge, once we're outside of that valley, these features will be really steep on the side closest to the ridge and then start to fall off and slope away from the kind of big volcanic features outside of that rift valley. Well, I do what I can.
There was apparently a bubblegum coral in this general vicinity that somebody wanted a zoom on.
And you have another outcrop, Scott, on the, if you look on the blue view ahead of you.
Oh yeah, off to that direction.
Yeah.
West, east, or southeast. Pilot, just before we leave, I think just down below us there might be some bubblegum corals, which are kind of thick-branched, reddish. I just walked in, so I wonder if it's that thing. I wonder if it's the bright white thing right in the center, branching, looks like it's sticking out of a sponge. I gotta go. Now it's just up above left of center, but I'm not convinced that's it. Oh, right on the lasers. Yeah, right below the lasers, kind of pinkish. Yeah, that's it. That's it. Okay. Yeah, that's definitely it. So here's a bubblegum coral with the polyps extended. Earlier today we saw one where the polyps were retracted. And you can see the kind of thick wads. These branches are really quite thick. And this one seems to be growing. I can't tell if it's actually growing off of that madrepora or if it's growing from behind the madrepora. But that's good. Thank you. It's another one of those Hertwiggia yellow vase sponges. Seen a few of those today. And then you can see that this is an interesting transition as we, thank you for leaving the lights on for a second. We're coming off of this, you called this like a, I don't know, pillow cone or eruption. What did you call it? Not a vent.
It was structurally, but not tectonically. It's just a ridge. But
okay, so this little ridge, but as
yeah.
it goes down onto this flatter part, you see all of those suspension feeders are gone, and we have accumulations of the rubble and so on. It's just neat to see that transition.
But in regards to terminology, that ridge word I'm using is not to imply any type of volcanic or tectonic activity. It's just same as a crest or just a
structural high.
Yeah.
Pilot, can we get the lasers back on, please?
Yeah. So looking at the bathymetry, I think that we have a decent more that we can shallow potentially.
I wonder if this is probably another big Stylasterin black coral fan in the distance there, and flanked by a couple of Leopathes black corals. They grow big around these parts. Lots of sponges. I'm not sure, maybe the madrepora.
So you were talking before about the branching on the corals, whether it was genetically controlled or possibly environmentally controlled, and I can't help to draw probably the very, very bad correlation to trees and how some fruit trees, where you clip them, it'll actually precipitate more budding and branch growth depending on the hormone feedbacks. Do you think that there's any similar phenomenon in the corals in terms of, the places where we snip corals, will you get significant more branching at those locations?
Let's have a zoom on the anemone as it goes by. It might not have to zoom since it's come to us. There we go. Thank you. I think it's actually a really good analogy. I would predict that there will be regrowth, and it'd be interesting to see if it's a normal regrowth or some aberrant branching, because, we did see in some of those past dives what we believe. No, we've imaged that really well. I mean, you can get just a brief close-up to verify that it's the same thing. One thing that would be nice, though, is this has a clear shot of the holdfast, that black disc that's attached to the rock at the base. If we could place the lasers on that, that will help us understand something about the size, and later on when we do get some aging of other corals, we compare that part of the colony. Thank you. Often you can recognize species of trees. A maple tree grows different from a holly, from a Bradford pear, that sort of thing. So there is some genetic control, or like a cypress tree. That's a good example. They're always straight and narrow.
And the canopies grow differently just by the variation of the tree.
But they're not all identical, right?
Yeah.
And so the branches go in different angles depending on how much sun they're getting. So maybe there's more branching on one side of the tree than the other. So I think that is what I mean about local control of hydrography versus genetics. And then on top of that, you're right, the clipping, because it's asexual and the tissue is still alive, can grow back. So one of the magnificent things about this black coral colony, I believe this is Stylasterin again that we're seeing, is you can see that it's playing host to dozens of comatulid crinoids, or feather stars. I also see a hermit crab on there. And it looks like they're all on the upstream side of this. So before we were told the current was coming across the ridge from east to west. We're kind of pointed almost due south right now, actually. Yeah, this is great. Oh, no, they're on both sides, the crinoids. Some barnacles in there. And I apologize because I was talking. You got the lasers on that holdfast? Yeah, thank you very much. Yeah. Yeah, the base. Yep. Excellent. What's that? After you finish with this shot, I want to go back in and look at what the yellow thing is right at the base to the lower right of that colony we were just looking at. See, it's got like 4 long, stringy arms. It's just zoom straight in, right in the center, and now it's to the 5 o'clock of the lasers. That yellow thing you see 4. I don't know if that's a black coral that there's 4 of them growing from the same position. Yeah. This yellow thing, that's what I'm talking about. Yep, exactly. Yeah, so we saw one of these before, and I thought it was in an unbranched form, but now I see that it branches from near the base. So that's great. Maybe we can get an ID from shore on what this thing is. Fernando, what do you think? You want the challenge of trying to clip this big blue colony, just get a little piece of it and suction that? Yeah, I mean, we've seen more than one of them, so we could hold out and hope we see another one if this is not a good location. So today we have seen orange-red black corals, purple-blue black corals, transparent black corals, and yellow black corals. That thing there with the what look like 4 strings or tentacles is yet another black coral.
But still haven't seen my favorite color.
So the yellow one is in the family Parentipathy. Okay. Yeah, we don't need to go far. So what I would say is a small piece that you feel comfortable can be suctioned. It should be relatively flexible, so go down the hose with no problem. But one thing about these is the skeleton is harder. It's more dense than what you've been clipping so far, so you've got to get it into those clippers well. What? Your survey one, yeah. Go for it. Telepathies with a Y. Yep. Can't say I didn't warn you that had a dense skeleton. It's impressive. Survey 1, this is watch lead 2. Request to put a small piece of this in formalin. Yeah, pilot, I feel pretty confident that this is flexible enough that it will fold up. In the canister. Very nice collection. Very nice. Those black corals are always tough. So an interesting observation being made by our scientists on shore that these really large colonies, this Stylasterin that you're seeing, are particularly susceptible to longline fishing. When the long lines come down and then they're dragged up, they get snagged on these really large colonies that are unfortunately right up at the top where they'd probably be putting the long lines as well. So another reason why we're kind of exploring some of these habitats and trying to identify what are the places that have these vulnerable marine ecosystems, these communities that are most susceptible to damage, and in particular where you have the very slow-growing ones that may take centuries to replace. It would be really good if we could cordon off that area and try to restrict the resource extraction from it to protect these magnificent ecosystems. We're ready to move on, I think, after that. Thank you. That was a really nice collection. Those black corals are always tough. My colleague noted that the yellow one is named Antipathes viminalis. That's that one that had the 4 long branches that branch from the base, that black coral. So, very colorful black coral day. Are you able to get a zoom on this large colony that's at the bottom center? Yep. This is a new one for the day. I'm wondering if it's another black coral for sure, and I'm wondering if it's the genus Stylasterin. As far as I know, there's only one species in that genus, Stylasterin magna, the magna referring to its size. The only other thing I can think is that some giant Parentipathies. So this will be interesting. Can you get the lasers on, especially on the base? Excuse me. This one's huge. There we go. So that's that's close to 10 centimeters across, just that black disc that you're seeing. That's the holdfast for this black coral. You can really see that shiny, smooth black skeleton that is otherwise underneath this tissue that we usually see. This is really cool. So the telepathies that I remember seeing do have some sub-branching, and I don't see any sub-branching on this. So this just might be the genus Parentipathy, and it's just a very, very old one. Very cool. Some of the polyps, look how long these tentacles are. I noted this yesterday with this particular family of black corals. The polyps are really narrow and they're stretched out along a branch. And so remember, as we're zooming in here, each polyp has 6 tentacles. And because they're so stretched out here, what you actually see are 3 pairs distributed along the length of a branch. And in the central pair, they will have the swollen body with the hypostome, which is the raised part of the polyp that has the mouth on it. I remember when I first encountered a Parentipathy, I could not for the life of me figure it out how the polyps were organized because of this weird stretching. And keep in mind, you know, in the middle of each 6 of these tentacles is a mouth. So, I mean, we're literally looking at thousands and thousands of mouths hanging out in the water column, I assume, all trying to grab something to feed. There's some suggestion that some of these corals may be able to feed on dissolved organic carbon, so some molecules that are dissolved in the water they may be able to uptake. I think that would require some stable isotope analysis of tissue in order to know for sure. That's a great view, and when video's finished, we're good. Thank you. So the consensus is that is a very large Parentipathy. Lots of sponges down here in this terrain we're going over right now. Our depth is 998 meters. This is the second one of these large kind of amphitheater sponges that we've seen. I've zoomed in on one of these before, and if we could have the look at the back of this large sort of dinner plate, the brown thing that's on it. Just curious what's sitting on the back of the sponge. No, I want to be on the top here. Yeah, I want it on the top side. It looks, from here, it looks like an anemone on the top of the sponge. Isn't that what the pink thing is in the center of the sponge? Oh, okay. Your eyes are better than mine. That's an Okeanos. Oh, what a fantastic view. So this is Okeanos, the bamboo coral, and I told you earlier that it could grow on a rock with a holdfast or in the sediments, and when it's in the sediments, its skeleton drips like wax. Well, here it's dripping like wax on the back of the sponge. So those things that you see dripping down there, that's the base of the skeleton, and that's what allows it to grow in the sediment as well. Oh, what a beautiful view. That is great. Yeah, nice stuff. Very cool. So that's all calcium carbonate that's been secreted by the coral itself. Fantastic. And if you see it on a rock, you'll just see that that's like a little round disc. And I think that brown thing just to the left, we're looking at the top view of a Parentipathy's black coral. Well, I'm glad I went and looked at that fake anemone. Everything, but no. No, we can move on, pilot. Thank you. So for folks unsure, we are now headed more or less north. So we are moving along the top of this ridge-like feature that we've been scaling for the earlier part of the day. Nice jellyfish there moving through the water column, maybe an atolla. I take it back. I don't think that's atolla. Beautiful view, beautiful diversity. I think there's another one of those large purple Stylasterin black coral in the background. Now I'm kind of doubting myself. I'm not sure if in the background just to the left are two bamboo corals or if those are those purple plexaurids that grow on the long stalk. I can't identify it from a distance anymore. Oh, for the first time, pilot, if you can just keep going left, that's good. You see that squiggly sort of line in the lower left corner? Yeah, please. That's probably a Phanosticopathes, another kind of black coral. Copy that. Nice job up there, Fernando. Yeah, the squiggly line there, that's the one. Yes, indeed. Another black coral. You can see at the base there where it's attached to the rock by its holdfast. It's missing tissue on the bottom a few centimeters, and you can see the black coral skeleton through the tissue. And otherwise, you see that all the polyps are aligned on one side. You can see their tentacles extending there. And we used to call these Sticopathies, but I think this belongs to the clade that has been shifted over to the new genus Aphanosticopathies. I think that was just published earlier this year. So good image of that. I think that this is the first one of these that I've seen today. Yeah, good stuff. So I'm ready when the video's ready. You can see a snail in the background there if you want to play with your focus. Or is that a hermit crab? A hermit crab. Yep, see his little toes sticking out in front. Couple more of the Oreosomatids, the Oreo fish drifting through here, urchins. Look at all the urchins here. So, Pilot, if I understood correctly, or Pilot in video, if I understood correctly a little earlier, there's a little bit of iris control problem. But if it could be lightened up just a little bit, if it... Thank you. Sure. Understood. Yeah, this was a comment from earlier when we were up against the rocks, but I totally understand.
Yeah, just to give a heads up, unless we see something unbelievably astounding and out of place, I don't plan to collect any rocks here.
And Nav, I was listening along there. That was accurate assessment, and we will want to do those last two Niskins, one just before we leave bottom and the other up in the water column. Lots of Okeanos colonies, these bamboo corals that are kind of orangey brown in color. I've seen them all over this flat. Oh, there's a new fish on the lower right at about 3 o'clock, 4 o'clock. New for the day. Let's get some good imagery of this one. Must be some kind of Sebastes scorpioniid or something like that. What was your suggestion? Yeah. Nice. Having a little chat with that cup coral just in front of it. Trachyscorpia cristulata, the Atlantic thorny head. Thank you, Telmo. It's Telmo Murado down in the Azores. So I can tell you we've got the fish ID'd, so you can do that for video, but we have what we need. Okay. Thank you. I don't mind video. I just wanted to let you know that. Yeah, usually we are. We, as you were saying, we need to get that good shot of the head, but I guess this is a well-known one. Seamount small. Oh, small parentipathies. I don't see it. Again, lots of diversity, really good abundance up here. You know, there was one point earlier today we were sampling, I forget what, and I saw a small rock pen colony, but I haven't seen any others since then. And you can bet I've been looking. This must be another Parentipathy's black coral, this feather-like thing that's sticking up right in the center. And that's just like the large one that we were imaging a little ways back. Lots of sponges. I'm seeing another one of the yellow Antipathies going off the screen to lower right. More Acanella bamboo corals, those light orange colored colonies. What's in the water column drifting towards center? I guess it's a fish, but I'm not certain. Oh, it's an Oreo. That's okay. Yeah, it's another Oreo. I should have realized the way the fins kind of wave there. We're good with the fish. Thank you. That feather-like black coral that I just referred to as Parentipathy's is apparently Parentipathy's pseudo-alternata, as the species name. And when there was a little bottle brush next to it, that was a Parentipathy's black coral. If you can, Pilot, in this overhang, the left side, I think I see something purple tucked under there, and I'm not sure if that's a fish or not. Do you see what I mean? Just in that little overhang. So underneath. I'm trying to find it. It's to the left of the lasers. The other side of this little grotto, there's something purplish in there right behind a little bit of a fan. So just to the left, and lasers on it now. From this angle, I'm now thinking it's just going to be rubble and not a purple fish. I was hoping it was a codling or something that was stuck down in there. Ashton, Les Watling is wondering if this is a carbonate crust. He saw a lot of the carbonate crust on the top of the cornerized seamounts.
Yeah, I think this is all partially lithified carbonate crust and carbonate ooze. You know, I'd expect there's some manganese on some of the harder rocks, but nothing more than maybe half a centimeter at the most in terms of manganese crust.
So would this carbonate be from coral reef, or is this consolidation of...
I think this is consolidation.
I see.
Yeah.
Well, I would suggest if we just do the quick zoom, I can tell you if it is something of interest to spend time on. Yeah, so it's not a fish. That's a Parentipathy's black coral, so that's fine. I didn't expect there to be corals growing underneath that ledge. Thank you. There's also a cup coral. Yeah. It's what? That's a cup coral. Yeah, there's two of them. One that's got all the tissue on it, and the other one I think might be dead. Still lots of these isolated Lophelia and Madrepora colonies. They're the ones that reflect pretty bright white or a slight beigey color. Those are the stony corals. I mean, there's so much in this view, it's hard to describe everything. Caitlin, our video clipper back here, was just asking me if any of my grad students have to identify all this stuff. And so sure enough, I have 5 students in Lafayette right now who are working on annotating these videos. 3 grad students and 2 undergraduate students. And that would be James Aubrey, Pashna Ganguly, and Mary Durie are the graduate students, and then Elena Hebert and Chelsea Hunt are the undergraduate students. And so, in particular, those undergrads have had a crash course this summer in identifying deep sea invertebrates as they prepared for this course, this cruise, excuse me. So, good work by them. And the email I've had from them, they're really enjoying this expedition. And don't feel bad for them. They're being paid for it. I can see, you know, in the background there, that's right now we're turned towards the northwest, which is kind of looking a little bit down the slope. You can see all of the stuff that is coating the rocks down there. Quite impressive. But we'll be turning a little bit to the right and headed north along this ridge, I believe. Wow, pick something. Let's see. Swing just a little bit. You're going to be going to the right, is that correct? Yeah, so if we just turn a little bit to the right. Let me have a look at what's on the rock here that we haven't had a good look at yet. Perhaps the yellow fan thing just in the center. Not even sure at this point if that's a sponge or if that's an Alanopsamia, which would be a stony coral. Who knows? Maybe it's yet another kind of black coral. Starting to think it's neither of those choices. Well, it's definitely not a sponge. Can rule that out. I had to get that out there quick before Ashton pointed that out to me. Cause,
that's like a biology joke, isn't it?
Yeah. So I think this is another black coral. For goodness sakes, how many colors of black coral are down here? And there's a whole lot of crinoids hanging onto it. I'm pretty sure, based on the pairs of tentacles that I'm seeing coming off of each side, that this has got to be a black coral. Yep. Wow. That's a really yellow one. Sako is transmitting information from Tina that is Parentipathies, she says, based on that yellow color. I got to keep a list here of all the different colors of black corals that I've seen today. That reddish brown thing that we're just pulling out that the lasers are touching, that's another black coral. And the bright orange one that's coming into frame on the right, that's another black coral. So you can see that the tissue can take on a diversity of colors. So is this feather-like thing in the lower left. They're all different species and mostly different genera, so they're not all that closely related, but they all are all in the order Antipatheria. Cool. Les Watling says black corals are anything but black. So clearly the first black coral must have been described by somebody who found the skeleton with no tissue on it. We might have been calling them all yellow coral. All right, we're ready to begin our move, our transit here again. Those sideroid urchins have been quite common today. See one tucked into the rock on the left. Some of our scientists are on shore are noting this, and I've seen this for a couple of different things now, that some of these corals that are very colorful, when we collect them and then we preserve them in ethanol, they turn black or they turn blue, and that's an indication that the pigment that is providing the color is in the soft tissue and not in the hard parts, whereas others will retain their color because that pigment is associated with the hard parts. Impressive view of that urchin. We've got a crab up above it, looks like a brachyurine crab of some kind. I personally am interested in that fuzzy white stuff that's on the upper right. That looks like the primnoid Thuarella again. And then arising from behind it is a purple plexaurid or Gorgonian sea fan. Yeah. Interesting. The last one we saw was also, or the last one I remember seeing, we've probably passed over others, but was also associated with this Madrepora, which makes me wonder if the larva is preferentially looking for you know, a calcium carbonate substrate that it wants to settle on instead of directly on the rock. Good stuff. Thank you. Cup coral down there. Does anyone on shore know those crabs? Give me a name. So, Pilot, I'd be happy if we, kept contact at the bottom, simply because there might be different kinds of fish on these flat parts. No worries. Understood. Thank you. Lots of Acanella colonies, again, dotting the flatter parts here. So these Acanella bamboo corals seem to have a preference for the flatter areas than the higher areas. I think I see some Chrysogorgia out there. There's that large purple plexaurid again. There's a couple of colonies of it on the left. Another one of those umbrella or hood or amphitheater sponges. Actually two. There's two of them on the left, and there was one out of screen now on the right. There's a request to zoom into the Gorgonia, so that would be the fan that's just going off the screen to the left. Carlos, my thought on approaching that was that it was the Anthothelidae, but we'll see what kind of a view we can get of it. Oh, wait, is that a fish sitting on the rock, or is that a rock?
I think that's a rock on a rock.
Oh, it is? Oh, I thought it was an eel. Oh, yeah, a rock behind the first rock. Got it. Rockfish. Now this is giving me more of a Candidella sort of vibe, which would be a kind of primnoid. I know I asked you to turn up the lights before. Can we turn them down just a tad? I think this is a bamboo coral, and it's just a little heated. Understood. Yeah, that's good. Yeah, I can see it now for sure. So this is a bamboo coral, almost assuredly in the J3 clade, probably Jasonisis, I can see that it is branching from the nodes. It's got a pretty classic, the high reflectance of the light is probably due to all the scales, scale-like sclerites that are embedded in the polyp wall and in the coenenchyme. Coenenchyme is the name of the tissue that joins all of these polyps.
Okay, back up for a second. What's a J3 clade?
There you go. You got me. So, Les Watling and my students and I have for more than 10 or 15 years now been examining bamboo corals around the deep ocean in the Pacific and the Atlantic, South Pacific, and been doing genetic analysis of them. And one of the things you can do once you get the DNA sequences is you can compile all those sequences into what they call an alignment to make sure you're comparing always the homologous or the apples to apples comparison of the sequence. And then you do an analysis and you build what's called a phylogeny. It's kind of like a family tree. And that family tree allows you to say which of the species that you're looking at are more closely related to each other and which are more divergent. And then that allows you to ask these really interesting questions about how certain characters evolved, have they evolved more than once, and so on. Each major branch on the tree that then puts out more branches is called a clade. And so we have not yet finished all the taxonomic work and the manuscript writing that's required in order to put names on those clades. And so temporarily we use just the clade name. So clade J3 is telling us it's a branch of the phylogeny, an evolutionary lineage that has a number of species that are all related to a common ancestor. We have at least one of them in that group that has been described, Jasonisis. And in fact, Les Watley and his student, just during the cruise while we were at sea, their latest paper came out, and there's a second species in that clade that has been described, and that is called Dodecaesis. And so we have two names now in that J clade, and eventually we'd like to have names on them all so we won't have to use that clade terminology anymore. It's a great question. I think that fish was in the family Moridae. Antimora, perhaps. I've been getting different common names for that. Antimora rostrata. The one I'm looking at here says violet cod. I heard, I think, blue Antimora yesterday. Yeah, absolutely. I think this might be a salp chain. And probably it's that or it's broken off from siphonophore. So if it's a salp chain, each of those three balls would be a pelagic tunicate. And they do grow in these chains. It's basically a colony of separate individuals. And they are filter feeders that have this barrel-shaped body. The bright white that you see would basically be their guts. And you can kind of see, yeah, on that angle, there are these filters on the inside, the sort of more gray-like structure. And you can see there's an opening at two ends. Beautiful video. Yeah, that's great. So that's definitely a salp. And so they channel water through the body, and you can see, oh, exquisite. That is great. You can see those filters that pick up the organics, this marine snow that's in the water, and that's what they feed on. And they can form these pretty long chains. So they're related to the sea squirts that you might see if you go down to the seashore. So yeah, it's got really close to us. And a few days ago, we saw a high-period amphipod riding around in a hollowed-out kelp. So there's the live kelp right there. There's the, yeah, let's check and see if this is indeed the Antimora. Sure looks like it to me. Yeah, yeah. I've seen several of them now. All, yep. Coincidence. Yep. We're talking here in the back row about that sponge, what I called before the beach umbrella sponge, growing over that coral. And my suspicion is that the coral grows faster, the sponge is growing faster than the coral, and so grew over it. But it's also possible that the larva of the coral settled on that rock and then grew up and discovered, oh, there's a sponge above me. Really nice shot of this Antimora. Pilot, these things I'm calling beach umbrella sponges, they're kind of like these big amphitheaters. There's an interest in getting a piece of that. What do you think? Nice video. And I imagine it's kind of and Pilot, just for context, we've seen lots of these sponges, so if this is not a good position for you, we can get the next one. Yeah, exactly. Cut a piece out. So I think this is going to be kind of like a texture of a stale cake. You know, it's going to be somewhat stiff, but mostly soft. So you're going to be able to break a piece. And I wonder if you go down to that lower lip on the right side, if that would be the easiest way to sort of get your manip in there and just break off a chunk. And then, you know, if it falls, then we'll just slurp it. That's fine. If it doesn't, we'll put it in the bio box. So getting back to that that fish we were just imaging, which was a morid, the information I'm getting from shore is that it's more likely Lepidion equis rather than Antimora rostrata. So still in the same family, but a different species, and that's based on the pointiness of the nose. The nose on this one was not very pointed. The nose on Antimora rostrata is quite pointed. Thank you for that input. I'm not certain what it is you're going to grab, but any chunk that's more than like 4 centimeters in a dimension is fine. I was just giving you a target to go for, just to grab a chunk. Yeah. That looks even softer than I thought. Certainly a demo sponge, not a glass sponge.
Nope. Nope.
Oh yeah, that's a great chunk. Wonderful. No, no. They're using microscopic parts of the internal part of the sponge for most of the ID, and along with the fantastic imagery we have, that'll be plenty. Thank you. Nicely done, pilot. Copy that. Another whip-like bamboo coral. So earlier we were looking at one and I was talking about, at what point does it start branching? So that's a whip right now, but maybe it's going to look like the one that we see in the far distance to the right in another 10 years. Curious to know what the signal is. So. Lots of Okeanos bamboos, cup corals. In fact, Shannon Hoy just sent me a nice little diagram of several different corals that maybe I can use to put some names on these cups. It's a yellow plexaurid. We did get a piece of a yellow plexaurid earlier. That's what's on the top of that rock that we just centered on. Just gorgeous. And there's the that really interesting to me, this yellow-black coral down there with the 4 long branches, and they all arise. Those branches all arise from the base. They don't seem to subsequently branch, so they often look like the arms of some brittle star or crinoid or something tucked down in the rock. Oh, can we have a look at that large urchin that's on the right side of that rock? The pincushion? Yeah, the pink pincushion. It's the first time today I've seen this kind of urchin. We've seen the sideroid urchins like the one in the lower left with those huge, massive spines. This looks like a more regular echinoid to me. Okeanos, I mean. So in general, the sea urchins are grazers, and so they would be moving over this hard substrate, and they have fairly complex jaws that they would use to scrape the surface. Down here, I would imagine that's bacteria, maybe settling larvae, very small sponges, stuff like that. You can see they have fairly long tube feet. I can see some of them waving on the top surface, but then if you look at the upper part, you see they look like lines that are securing them to the rock, and that's probably exactly what they're doing. You see they're really straight, and the bottom of the tube foot is, I think now they say that there's a chemical that acts like a glue, not that it's suction. You can also see them all along the left margin there. That's great. And you see the other ones waving. You can see the sort of white tips to those tube feet. Really nice view. Manuela Ramos is saying Okeanos esculentus is possible. I don't need a closer shot of the urchin. I don't mind if video wants it, but I'd like to get a shot of that coral, the fan, that's just to its front left. Yeah, just slightly below it. Yep, so in the same position. We've got interest from a couple of different scientists on shore about whether, well, exactly what this is. One suggestion was that it was a bramble bamboo coral, so I'm looking closely to see if I can see nodes in the skeleton. To me, it doesn't have the structure of the bramble. It's too planar. See how close we can get here. Wow. We'll keep our eyes out for another one of those. Yeah, there's a lot of interest because it's been fairly frequent and people aren't sure what it is. So we'll go back in. Thanks. In fact, I think there's another colony of it that's almost directly under that pink pincushion and slightly to the right. So on the rock face that's facing, yeah. So if that's an easier one to get, I think they're the same thing. Yeah, I think. Okay. Yeah, we're going to have to get really close. Yeah, I don't think it's a bamboo coral. I really don't. That's good. Well, I'm stumped. I'm not sure what it is. Orange coral, maybe Paramercia. I don't think I've been this quiet all day. I don't know what it is.
A taxonomist hard at work.
Yeah.
I went online. I wanted a good definition of what taxonomy is. I think the best one I could find is, and correct me if this is not right, but it's the study of organisms and how you file them.
And how you what?
File them.
Thanks. That's good. But if you had a drum, so just before we go, somebody else has called out a yellow. I'm not sure what they mean. Zoom on the orange coral. We think it may be a Paramercia. I'm not sure which one they mean. No, those are zoanthids. I mean, we could look at those, or maybe that's Iguchisamia. The yellow thing, so who's asking that? Marina. The yellow thing in the background there, that's a black coral, an Antipathy's black coral. We got that ID'd earlier. Oh, she's talking about the very top of the rock. My bad. That's what it is. No, on top of the pincushion, at the very top, that orange fan. Yeah. Yeah, that's almost assuredly Paramercia. No, just above. It's gone off the top of the screen. Yep. The king of the hill. That's the one. Yeah, this is, to me, this looks very characteristic. The family Plexauridae, genus Paramercia. It's a very common genus in this family in the Western Atlantic, also the Eastern Atlantic, actually. Lots of different species, and very hard to identify the species by video. Perhaps they have an ID based on its locality. One thing I find interesting is there are brittle stars. Okay, I was going to say there's often brittle stars or snake stars associated with these colonies, and you can just see the back of the arms, so the body of the snake star must be on the other side of this. There's at least one anemone growing on it. So that's good. Yeah, I would agree, Carlos. I think this is different from the one that we sampled earlier. All right, let me just relay to shore. 30 minutes left, or now somewhat less. I'm going to put 25 left of bottom time. All right, the message is relayed. So the question is, do you want to continue along this ridge to see, how much of this community is there up here, or do we want to continue zooming? We really don't have much in the way of collection space left. All of our bio boxes are full, and we have 2 open suction canisters. And about 10 hours of sample prep in the lab after with all these associates. Oh, my goodness.
I don't think you were looking at me. I think you were looking past me at Harvard.
You're going to be busy with the rocks tonight.
Oh, I know.
Let's get a nice look at this purple colony that's coming in the top left. So this is one we did look at earlier. This may be what we sampled, although it didn't have this fan-like morphology, but it could be. And you can already see from a distance here, it's got a combination of both the purple and the yellow. And I can tell you that in the canyons along the east coast of the U.S., Norfolk Canyon and Baltimore Canyon, I've sampled Parentipathy that range from purple to yellow and are mixed. And so we found all of those within one genotype. They're a lot shallower than this. It's between 300 and 500 meters depth. Yeah, gorgeous. So again, I think what's going on here is we have purple sclerites. These are the calcium carbonate hard parts that give the main stems that purple color, and then we have a yellow pigment that's in the tissue that gives the polyps their color. And so if these polyps were all retracted, this whole colony would look purple, but because they're extended, you get that mixture of color. Here's another one of these colonies that we were just imaging close up before. Underneath it, that we still don't know what it is, and there's all kinds of discussion raging in the chat room about it. And Les Watling had another hypothesis that it was the genus Dendrobrachia, which I think would be really interesting. They're an unusual octocoral that has a bit of a spiny skeleton. I've never actually seen one live, which would explain why I couldn't identify that one. So that would be very cool. All right, thank you for that. And when video's happy, I'm happy. I'm reading back through the chat room. There's so many messages coming in, it's hard to keep up. So I'm trying to make sure I haven't missed anything really critical. Asako Matsumoto is saying that, or translating from Tina, that perhaps there were selenogasters around, and I certainly have seen selenogasters. Those are the aplycophoran molluscan worms that feed on the corals. I haven't seen any recently, but definitely today. And we did collect a couple of specimens, I think on the first dive, pulled them off of one of the corals. Carlos says that orange fan that we were looking at closely before, the one we suggested was Parentipathy, is pretty common here around the Azores in these depths. So there seems to be some consensus developing that that was dendrobrachia. How cool. Understood, pilot.
And so the co-pilot and the ROV pilot are just swapping positions. So we're just kind of hovering here for a second.
So back in a flatter area with, now I'm not sure if this is all coral rubble or if this is pteropod rubble. If it's coral rubble, how did the coral rubble get here? Is the current so strong that they're being washed from those little highs that we're seeing? Sure, yeah, let's just determine what this is. And again, you know, these Okeanos bamboo corals really seem to like it in this flat area. So let's see here. Those look like, I think, a mixture of pteropod shells and urchin spines, maybe? And some other broken up hash, I guess? Interesting. Thank you. Is that correct, what I wrote? Yep. Yep.
I can't help but be looking for more weird track marks like we saw the other day.
Yeah, no, I think that's a good idea, although the paper and where we were, they were all deeper than 2,000 meters, so they would be new up here.
Yeah. My mind's going to be unsettled until I get an explanation for those.
I think this morning I saw somebody, I don't know if it was in an email or if it was in the chat, that they were more convinced that there was something akin to beaked whale feeding, where you've got a cetacean that just sticks its nose in. But 2,500 meters, it seems pretty deep for one of those whales. But the shape, it seems right for that, you know? Yeah, have to be a very precise beaked whale. Maybe it likes what? Yeah. I'm seeing straight ahead, just before the rock pile, something sticking up like a feather. It's black from the bottom. Yeah, what is that? Thank you. Probably a Parentipathy growing off of one of those little cobbles. But just in case it's a sea pen, we'll have a close look. Yes, indeed. That is a Parentipathy black coral growing on that little cobble. And you can see the difference between this one and those Okeanos colonies. The Okeanos can grow in the sediment, where these ones have to be on a hard surface. So the larva here found this little cobble and was able to grow on that. And then that must have attracted these two little squat lobsters that you see very commonly on these Parentipathies. I think if it hasn't been proven yet, I would be willing to wager that there's a species-specific relationship between the species of squat lobster that's on Parentipathies and the species of Parentipathies. And there's a little hermit crab right at the base. No, we can move on. Thank you.
Well, here's the title of the paper: Geomorphological Evidence of a Large Vertebrate Interacting with the Seafloor at Abyssal Depths in the Region Designated for yada, yada, yada. And it talks about whale marks at a depth of 4,200 meters.
Wow. What kind of whale marks are these?
Let's see.
Cuvier's beaked whale. Yeah. Let's have a look at this large colony that is coming into view on the center. This is a nice one. This might be, I'm going to guess from a distance that this is the bamboo coral we saw earlier that we suggested might be Ecnomyces, but this is a much bigger colony of it.
Yeah, they talk a lot about beaked whales in this paper.
So does it say they saw them? I mean, or is it just conjecture like the rest of us?
I think it's conjectural, and they saw some whale acoustics on the sonar, but I don't think they'd identified.
Sorry, Nav, were you speaking to me, Watchley? Thank you, Nav. Yeah, I would like to get a tighter view on the branch in the front is fine, just so I can look at the polyp level. Thank you. It's got one of those, what was it? You had looked up the name.
No,
Stenodactyl, something like that?
Somebody had put the name in the chat, yeah.
Right, and then you were suggesting the steno referred to the hooks on the back, I think. Yeah, so in this case, all of these sternostyles, yeah, the polyps are extended, and so I can't tell if they have that volcano shape when they're retracted. But yeah, that's good. Let's look at the branch in behind because we got more of a side view to the left. Thank you. Yeah, great. So you can see some of those white lines that are moving through the polyp body wall. Those are the sclerites that are embedded in there. And what we can see is that they kind of curve up the axis instead of being straight up and down or perpendicular to the axis. And the other thing I am seeing is there are not really long, prominent needles sticking up through and around those tentacles. So that's all useful information. Let's see what Les Watling has to say. He's still talking about the beaked whale. Everybody's talking about the beaked whale marks at 4,000 meters. All right, thank you. That's great. So getting back to the clade idea, Ashton, that's definitely a declayed bamboo. And so it's either going to be a keratoisis or a ecnomysis, and that's what we were looking closely at those polyps for to see if we could tell. And I don't think we've done this all expedition, or certainly not this dive, but on the face of the rock, right in the center right now, is I'm pretty sure that's a brachyngid. And we haven't looked at any of those yet. That would, if so, that would be another echinoderm. This would actually be more closely related to the sea stars. I'm hesitating now to make sure it's just not a crinoid with its arms way outstretched. Nope, I was wrong. That's a crinoid. Okay, I can tell it's a crinoid because you can see underneath it those thin white lines. Those are the cirri, the feet-like structures that are holding it to the rock. What's nice, though, is we're getting a top-down view, and that dark slit that you see in the center would be the mouth. And then on the right side of that, using the volcano theme, there's a little raised cone, and that's the anal cone. And so because of the shape of the body and the way it sits on the bottom, both the mouth and the anus open up on that same side, and it's curled, and so the anus is raised up on a cone so that it isn't basically defecating into its mouth. It's a way to keep them separate. Oh, and there's another example of the dripping down waxy holdfast. I assume there must have been an Acanella in there at some point. Look at that. Cool stuff. Yep. That is very interesting. There we go. Yep. So I'm not sure, before when we saw that it was coming from an Acanella, I don't see an Acanella here. Maybe that's coming from the Lophelia. That's good. We're done with this rock. Thank you. Pentametocrinus is the word we're getting on that crinoid. Copy that. Oh, there's something of interest to look at. The problem is choosing, right? Let's go for that little bushy white thing just above the spiky ball in the center. Thank you. This will be our last close-up, gang. We'll see if this is anything different. Let's been a pretty successful day, if you ask me. Okay, this would be very interesting if we found something new right at the end of the dive. I'm kind of sort of wondering if it's a hydrozoan. Thanks. I think this is what we were looking at earlier that stumped us, and I'm equally stumped again. So that's a perfect way to end. Just to say that there's still some mysteries down here, some things to be collected, and that's great. So it's definitely an octocoral. I'm going to guess maybe, I don't know. I don't even know if it's a plexaurid. Seems to have, the skeleton doesn't really strike me as plexaurid-like. Yeah, Carlos is saying, yeah, they're wondering the same thing. Nobody can place this. Well, I think we're out of time. Yep. Don't forget the Niskin before you leave the bottom, huh? Thank you. Thank you. We just took another water sample before we leave bottom, and we will do one more when we move up in the water column back through the deep scattering layer, and so that'll give us 5 water samples for eDNA for the day. All right, got my shore, got the databasing done there. All right, folks, so that's bringing to an end our bottom portion. In just a couple of minutes, we'll have a discussion about tomorrow's dive. I think that in the email I said 4 o'clock, and if that's the case, maybe we should wait until 4, just in case anybody else who's not currently on the dive is planning to dial in. I will check that. Yeah, I said 4 o'clock local, so that's about 10 minutes from now. So those of you who are interested in talking about our dive tomorrow, which will be further to the south in the Kharchov fracture zone area, please stay tuned, and we'll discuss that in 10 minutes. Otherwise, I really appreciate your spending Sunday with us today. It was a pretty spectacular dive. I'd say this is a place worthy of protection based on what I saw. And we got a lot of samples collected, and we'll be working for a while processing those tonight.